My name is Vanessa Ruta, and I'm an assistant professor and the head of the Laboratory of Neurophysiology and Behavior here at Rockefeller University. And I'd like to welcome you all and thank you for coming to this event this morning. We have a record-breaking number of students and teachers at this event today. There are 70 schools that are represented, and you've come from Manhattan, the Bronx, Brooklyn, Queens, Staten Island, Westchester, Long Island, New Jersey, and Connecticut. We are very pleased to welcome several schools that have not been at this event previously and who are new to our campus. I'd also like to extend a really special welcome to those of you who attended this event as a student and actually have now returned as teachers. We'd like to believe that your experience here at Rockefeller actually had a meaningful impact in your decision and encourage you to pursue a career in science. So the Rockefeller University is one of the world's leading biomedical centers. It was founded 114 years ago, and it's actually been on this campus for over a century. In that time, the university has created a culture resulting really in an unparalleled record of scientific achievement. Scientists associated with the university have actually won more Nobel Prizes in chemistry and medicine than any other institution in the world, 24 to be in fact. So if Rockefeller University were a country, we would rank 4th in the world in terms of Nobel Prizes after the United States, the United Kingdom, and Germany. Scientists in our 75 labs conduct research in a wide array of different disciplines, conducting innovative studies really aimed at improving the understanding of life for the benefit of humanity. So to really give you a sense of what makes Rockefeller so special, I'd like to now show you a brief video about the institution.
Rockefeller University was formed in 1901 by John D. Rockefeller Sr. And the reason it was formed was that we had reached the point in the state of medicine where you could diagnose what was wrong with people, but we had no ability to stop the disease that they had from killing them. Rockefeller had an advisor named Gates, and so Gates came up with the idea of bringing together the best and the brightest in one location to basically study how do we interact with disease and how do we stop disease.
My own laboratory studies a rare group of patients who, amazingly enough, have an immune response against their own cancers. We're able to discover something that people haven't seen before, which is human beings have a natural ability to get rid of cancer. Not only do the best scientists want to come here, but the best students, the best fellows, the best clinicians want to come work here. This is the #1 place in the country, possibly the world, to do this kind of research. So I'm privileged to have an absolutely spectacular group of people working with me in the laboratory now and hopefully in the future.
Rockefeller is important to what we're doing because it's one of the only places in the world that actually prizes risk-taking and has allowed us to do completely improbable scientific experiments that we just wouldn't be allowed to do at any other university. Mosquito disease transmission is an enormous problem in the developing world. Malaria kills 1 million people a year. Several hundred million people are sick and infected with the malaria parasite. To try to find out what that substance is that's either attracting or repelling mosquitoes, and that substance, if bottled, could be an incredibly important tool in preventing mosquito disease transmission.
Rockefeller is just an incredibly exciting place to be because of the strength of the basic sciences on the one hand and the commitment that's always existed here to taking that knowledge and translating it into medicines that can help patients.
We need to identify those with passion and with the intellectual capabilities to back up that passion to go into medicine and science.
Biomedical research is our only focus when we're here, as opposed to being in a large medical center where often your research competes with the time that's spent in clinical care. Rockefeller is second to none in providing that protected time. Obviously, I'm very thankful for the investment that Rockefeller has made in me. As part of the Clinical Scholars Program, I had 3 years of protected time. that gave me enough time to focus on my research, generate data. Now I'm an NIH-funded investigator, and that was not a small accomplishment, and I could do it thanks to the investment that Rockefeller made.
Rockefeller University is very unusual in that it is not divided up into departments reflecting different disciplines, because departments can form barriers between different sorts of research workers. What we have here, no barriers between the basic researchers and the clinical researchers.
Having no departments is critical. Every head of lab, tenured or not tenured, reports to the president of the university, period.
This is really the golden age of disease research, translational medicine, and drug discovery, and I think that medicine is going to be transformed profoundly in all of those different areas.
So I'd actually like to take a few minutes to tell you a little bit about my own path into science, which had some unexpected twists and actually was shaped by some very important experiences, including lectures like this one. So I was actually a high school student in New York City, just like many of you, and I attended Stuyvesant High School for Math and Science, which would seem an ideal setting and an ideal foundation for someone who is aspiring to be a scientist. But I actually came from a family of artists, and as a high school student, I really wanted to be a professional ballet dancer. That was my dream, and to have a career in the arts. And for 4 years after high school, I actually pursued that dream until eventually I came to the realization that I actually would never be quite as good a ballet dancer as I wanted to be. And as I was pondering my next steps, what came back to me is actually my high school chemistry course, which was taught by Dr. Matthew Litwin, who still is a very important figure in my life, because he really revealed to me just how beautiful and elegant and logical the basic principles of chemistry were. And so based on this one course, actually I decided to pursue a degree in chemistry at Hunter College. And when I was an undergraduate, I was very fortunate that I had the opportunity to attend a lecture, much like this one, taught by a Rockefeller, or provided by a Rockefeller faculty, Roderick McKinnon, who is now a Nobel laureate in chemistry, and who explained with his great enthusiasm and passion his work visualizing proteins that are really essential to the fast electrical signaling of the nervous system. And actually really inspired by that one lecture, it compelled me to come to Rockefeller to pursue my graduate studies, to get my PhD and work with Rod in his lab. And during that period of time, I really became to appreciate that many of the aspects actually of being an artist, the things that really compelled me and fascinated and excited me the most about being an artist, are really the things that actually make science such a wonderful profession. There's this incredible thrill in the creativity and discovery process that you feel in both of these fields. And I'm reminded really daily of the beautiful synergy between art and science. So after postdoctoral studies at Columbia University, I was fortunate to have the opportunity to come back to Rockefeller and begin my own lab 3 years ago. My lab studies how sensory information is processed within the nervous system, and we study the sense of smell or olfaction, one of the many senses that will be discussed in today's lecture. And we study how odors in the environment actually are being processed in a rather potentially unlikely model system, the fruit fly Drosophila melanogaster. And it's partially because these little insects actually perform quite a large array of complex behaviors driven by odors in their environment. They use odors to find the appropriate sources for food, the appropriate mates, and to avoid predators and other threats to their survival. And they perform all these complex behaviors using a rather simple nervous system, whereas each of our brains has over 10 billion nerve cells. The fruit fly has only 30,000 nerve cells in its brain. And that simplicity really allows us then to start to resolve the neural pathways in the brain that process information at a resolution that we could not attain by studying more complex animals like ourselves. I will be on hand today with some of my students to provide a demonstration of how we study the nervous system of the fruit fly. And I'm very happy to talk to any of you about the work that we do in the lab or my path to science at the lunch break. So it is now my great pleasure to introduce, and it's really an honor to introduce Dr. A. James Hudspeth, who will lead our exploration today on today's topic, making sense of the senses. So Jim is really a renowned sensory neuroscientist. He's the head of the F.M. Kirby Center for Sensory Neuroscience here at Rockefeller. It was founded in 1997. The center explores the biology of sensory perception and the disorders that can impair vision, hearing, balance, and the other senses. Jim's really beautiful work on the auditory system has begun to shed light on the mechanisms of normal hearing and are also showing how exposure to loud sound damages the ear's sensory receptors at the molecular and cellular level. Jim is a member of the National Academy of Sciences and an investigator of the Howard Hughes Medical Institute. Jim is also a fellow of the American Academy of Arts and Sciences and really a recipient of many, many numerous prizes. I'd say Jim is also renowned on campus actually for his incredible eloquence and being an amazingly effective communicator of science and an amazing teacher. So I think we're in for a real treat this morning. You may be also interested to know that Jim claims that he was born to be a scientist, unlike myself. Indeed, his first exposure to science was growing up as a young child on the outskirts of Houston near a bayou, where he started collecting pets, fossils, and minerals from a young age. And by the time he left for college to attend Harvard University, he had an impressive collection of over 200 pets, including a breeding colony of more than 100 box turtles. So that's clearly more than any New York City apartment could ever handle. So now please join me in welcoming Jim to the podium.
I actually don't do podiums, so here I am. I wanted to start just by placing sensory responses in a context, and this context is the context of a visit to the doctor's office. When you go to the doctor's office, you get asked embarrassing questions about sex and drugs. And after all of that, the doctor pulls out a little hammer like this, and she waves it in your face and begins to look for the tendon. And if things work right, you have a twitch when you're hit on this tendon. And you've got to wonder, what is this all about? I mean, in daily life, except maybe at lunch today, nobody ever comes along and taps on your knees. It seems a peculiar thing, but there's a real purpose to this. This is getting at a particular sensory response that's part of an important reflex. And this is the reflex that actually allows us to stand upright. When you think about it, just standing still is actually something of a task. If you have a mannequin, say from the store window of Macy's, and you try to stand it upright and let go, it'll always topple over. It's very hard to balance something as tall and narrow as we are, unless you're a ballerina. So what happens to the rest of us is we are constantly swaying, even though we don't ordinarily notice it. So as I stand here, I'm subtly, I hope, swaying forward and swaying backwards. And as I do that, this tendon in my knee gets bent as I go backwards. That elicits a reflex response that pulls me forwards. And similarly, when I go too far forwards, there's a reflex from muscles in the back that pull me backwards and keep me upright. So what this test is really doing is just a quick way of checking that some of that circuitry is working properly. So I want to discuss that circuitry as sort of an entree into the sensory receptor that's responsible for this particular reflex. This represents the knee jerk reflex that I just showed you. We've got bones in the leg. We have a muscle, this is the quadriceps, that stretches its tendon over the kneecap. So what happens when we elicit this response is that we're tapping on this tendon. That stretches the muscle, and that triggers a so-called muscle spindle, which is the little receptor organ that's responsible for the reflex. That organ looks something like this. It is a cylindrical object, about 1 millimeter or 2 in length. It contains about a dozen muscle cells, such as this little green cell that I'm showing you here. And these are specialized muscle cells. They don't contract appreciably to do any work, but they are sensory muscles. What makes them sensory is that there's a nerve fiber running from near the spinal cord that sends its ending to this muscle spindle, so-called, and wraps around it. And that makes this nerve fiber sensitive to stretching of the nerve fiber, of the muscle fiber, when one applies a stimulus. So if one taps on this tendon, stretches the muscle, one stretches this, that excites the nerve, and you get action potentials, that is nerve spikes. So here we have the system at rest. There's very little tension in this. We have only a few nerve spikes, pop, pop, pop. But as soon as we stretch it by applying a mechanical force, tapping on the patellar tendon, the number of spikes goes way up, as long as you continue to stimulate. So this is a very simple sensory response. The sensory response has 4 components that I'll be talking about in the rest of the lecture. The first is capture of energy. Somehow, something in the external world needs to trigger the response. So the stimulus in this case is a mechanical stimulus, a force applied to the patellar tendon by a little hammer that is transmitted to the muscle spindle. The second thing that happens is what's called transduction. Transduction in physics refers to the conversion of energy of one sort into energy of another sort. So if I hold this up, it has a certain amount of potential energy. If I drop it, which I won't, you will see kinetic energy, and that's a form of transduction. In sensory transduction, we're referring instead to the conversion of some sort of stimulus, light, sound, smell, or whatever it is, into electrical responses, which are then the common currency that's used throughout the nervous system. So in this case, the mechanical force that's applied stretches the membrane of the cell. That membrane stretch then opens an ion channel and allows ion current to flow into the cell, setting up a response. And I'll come back and talk at greater length both about ion channels and about the flow of ionic current through those channels. The next thing that happens is amplification. The amount of stimulus energy that goes in is usually very small. And in fact, we want to be sensitive to the very smallest stimuli that we possibly could. There's a real selective advantage for hearing the faintest sounds you can, for smelling the faintest odors you can detect, for seeing the most difficult to see objects. Because obviously the animals that can do that the best will tend to thrive and to replace animals that are less effective. So a tiny stimulus needs to be amplified. It can be amplified in different ways, such as allowing a lot of ions, which are separate particles, to flow through a single molecule, which is the ion channel. And the last thing that is occurring is called encoding. Somehow this flow of excitation has got to be changed into a signal that's sent to the nervous system. And that signal, as I've already showed you, is represented as firing of action potentials in a nerve fiber. So the louder the stimulus, the stronger the stimulus rather, the more firing occurs. There's usually a spontaneous rate, pop, 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 pop. There's a threshold, a minimum stimulus beyond which something begins to happen, and then the stimulus produces larger and larger responses, pop, 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 and finally, brrrr. So you will find this again and again in all the senses, and I will now walk through a few senses to show you how some of these particular things occur. Here's a schematic diagram showing that there is some input, whatever it happens to be, energy capture, transduction, amplification, and encoding, and then finally the output that's sent on to the central nervous system. Now there can also be modification of these responses along the way. So if we go back to the muscle spindle, here is the spontaneous firing rate. If we stretch the muscle very strongly, in other words, if I put my knee under a lot of tension like that, there'll be a huge amount of firing up here in the nerve. And I won't be sensitive to slight additional changes in the flexion of my knee. So the system adapts. It turns down its response through a process of adaptation. And the way it does it, in fact, is that this portion of the muscle and this portion both relax. So as they relax, it takes tension off of the central part, that takes force off of the nerve fiber, and it allows the firing rate to fall to a lower level. And this is, again, entirely characteristic of all our senses. You know, for example, if you go into a cafeteria, you initially smell whatever food is being prepared, but within a few minutes you cease to notice it. Your olfactory system has adapted to the odorant molecules. So we can show in this schematic diagram that in this case, energy capture and transduction is followed by feedback, a negative feedback that turns down the signal and makes a smaller response. You can also do just the opposite. There might be times when you're sitting in such a way that the tendon is quite relaxed and there's not very much spontaneous firing. The system is very silent. So now a very small tap won't be detected because it's below the threshold. So one can make the system more sensitive by the process of sensitization. And in this case, that's done by firing a different nerve. This is called a gamma motor neuron. It's a nerve running from the spinal cord down to the muscle. It causes a contraction to occur here, and in fact up here. And that contraction does just the opposite of what adaptation does. As this region contracts, and this region contracts, the middle region is stretched, and that activates the nerve. The nerve is now firing some spontaneous spikes, as you can see, and it's now more sensitive if you tap on the tendon or do something else. So in this case, we've done sensitization. We've accomplished sensitization by having a nerve fiber that affects the input going to the beginning of this cascade. So what are the senses I'm talking about? We have 5 classical senses, you well know: vision, hearing, touch, smell, and taste. And we'll be mentioning all of those. But we have a lot of other senses that we never really think about, largely because the output of those senses is going to our brain in a subconscious way. The senses are working all the time. They have a very important effect in our lives, but we just don't notice things are happening. So for example, our blood pressure is constantly being measured by something called the aortic body. It's a little cluster of nerve cells that are along the aorta where blood flows out of the heart. The CO2 concentration and oxygen concentration and acidity of the blood are all measured in the same place and in something else called the carotid body, which is up here in the carotids, the arteries going up into the head. So our sense, for example, of being suffocated if we're in a very tight environment comes about because the accumulation of CO2 triggers these receptors, the CO2 receptors, and that sets up a reflex that makes us breathe faster, it makes our heart beat faster, and so on, and it also gives us the oppressive feeling that being smothered tends to give. We have receptors for linear acceleration and angular acceleration. So linear acceleration includes, for example, the acceleration due to gravity, but it also includes any straight line movements that we have. If I close my eyes and move around in a particular trajectory, I can measure where I've gone, I can integrate that motion, and I can come back to where I began. And that comes about because of the fact that in the utricle and saccule, 2 organs of our inner ear, we're constantly measuring accelerations, including those due to gravity. Angular accelerations, again, are useful for football players and ballet students. If you rotate your head and body, there are another set of receptors that measure in all 3 axes of the body which way and how fast and how far you're rotating. And again, we are constantly using that information from both of these systems to keep ourselves upright. The next set are pretty obvious. You know what itching is. You know what pain is, which can occur owing to stimuli in the skin or in the viscera. Our skin is also sensitive to hot and to cold. We're sensitive to muscle tension, and that's what the muscle spindles do, among other things. And we're sensitive to joint position. We have receptors that are constantly measuring where our joints are in space. So if you close your eyes, you can tell that your arm is extended and your wrist is flexed and so on because of those receptors. And then there's a wealth of other types of sensitivity that we don't have, and some of it is really quite striking. So for example, there's infrasound, sound at frequencies too low for us to hear. So it turns out that wind blowing through trees or blowing around mountains produces very low frequencies, and some animals can use those to home. For example, birds can use them for migration as a consequence of the fact that they can hear these very, very low tones. Elephants can signal each other over immense distances with sound at frequencies much too low for us to detect. Ultrasound, many of you have heard of, used by bats and whales for hunting. And this again is a very sophisticated system. Bats, as they fly around, are constantly emitting little tones. And of course, we can't hear them ordinarily, but to more or less quote, they go, eep, 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 eep. As they get closer and closer to targets, they speed up their frequency of stimulus and get more and more information from what they're looking at. And there are 2 really neat things about what the bat is doing. When the bat goes, eep, makes a constant tone, it's using Doppler shift. So you guys mostly have heard about Doppler shift. If you hear a police car or an ambulance go by, you hear the frequency change as it runs past you. And this is because as it pulls away from you, the sound waves have to, in effect, travel at a lower speed to get back to you. So they're shifted to a lower frequency, right? So the frequency goes up as the thing moves away. Now imagine if you're a bat and you go, eep. If there's an insect flying around, by listening to the reflection from that insect, you can tell if it's approaching you or flying away from you. And as bats fly, they're constantly doing this, using their voices to detect where insects are, but also how quickly insects are moving so they can close in on them. Whales probably use something similar when they're hunting fishes and the like underwater. There's magnetoreception. Migratory birds in particular can sense the Earth's magnetic axes. So they orient themselves in space, and it's been shown that they can get to their targets based just on that sense. Sea turtles also seem to use magnetoreception, something else we don't have. There's something called remote thermoreception, remote heat sensitivity. Rattlesnakes and other so-called pit vipers have this. They can sense the heat of a mammal, and they therefore can hunt at night just by detecting the warmth of their prey. And this is something that happens to a lot of people who collect snakes, as I used to do. Didn't happen to me. But if you catch a rattlesnake and put it in a bag, which looks like a pillowcase, that's the way you carry snakes around, you feel very confident you can't see the snake. You go wandering off with carrying the snake back to wherever you're going to go, and then you get bit. Because the snake can see your leg through the bag. It senses the heat with its remote thermoreception, which makes sort of a crude image of the external world based just on heat sensation. So rattlesnakes are biting people all the time, experienced people who ought to know better just because they forget the fact that the snake can see something that they can't. One other sense that's not on the list will be demonstrated today during your lunchtime, and that's electroreception. There's some fishes that emit electrical pulses, just the way the bat emits sound pulses. And they use those electrical pulses to feel the environment around them. These are fishes that live in very cloudy water. They can't see well at all, but they can hunt based upon these electrical pulses that are constantly going out. So during the break, I hope many of you will see that. Vanessa's demonstration will be on the second floor of where you're eating, and this one will be on the third floor, but she'll give you more detail later. So let's turn to these specific functions, and I'll discuss them one after the other, beginning with energy capture, what I call the antenna function, the ability of senses to capture whatever the relevant stimulus is. One premier example of that is the photoreceptor. In our eyes, we have something like 100 million photoreceptors, rods and cones, that capture photons of light and convert them by transduction into an electrical response. So this is one such cell. It's called a rod cell. It makes us sensitive to light at low levels. So, for example, during moonlight nights. The light actually comes in at the wrong end of the cell and passes all the way through it to this so-called outer segment. The rest of the cell is ordinary. It has a synaptic terminal where it communicates to other nerve cells. It has a soma, or cell body, that has a nucleus. And it has a so-called inner segment, which has a lot of energy-producing organelles like mitochondria. But the key thing is the outer segment, which contains about 1,000 membrane discs. So these are stacked up like dimes or quarters in those little cylinders you get from a bank that hold 100 or whatever it is, nickels, dimes, or quarters. And each of these membrane discs, in turn, contains about 1 million molecules of something called rhodopsin, which is the special protein that captures light rays and helps to convert them into an electrical response. Now, as you know, there are 3 primary colors. We are sensitive, and you'll see this in a minute. We're sensitive to red light, we're sensitive to green light, and we're sensitive to blue light. And mixing lights of different colors produces all the other intermediate colors, including white. Of course, the absence of any of these gives the sensation of black. And the way in which our visual system works is very close to the way, say, a television camera works. If a television camera looks at this particular tree, the particular portion of it, say the tip of this little green branch, will be represented on a screen by a bunch of pixels in which only the green is illuminated, the red and the blue are not. So this represents a green object. The rest of this, with all 3 sets of pixels illuminated, represents the white background. And similarly, in our own eyes, we have the 3 types of photoreceptors. These are cone cells, red, green, and blue sensitive ones. In the region where the image of the pine tree falls, there will be photoreceptors of only the green type being activated. Where there's white snow, you will see red, green, and blue activated equally. So in this case, we have filtering that's occurring before the energy capture. We have different photoreceptors sensitive to different colors, red, green, and blue, and those filters are then applied to the input of each of the photoreceptors in the system. Now, I want to do a demonstration of this, and the demonstration involves what you just saw, the mixing of light of different colors. I'm going to mix together some red and green light to make yellow light. And the way in which I do this turns out to be a bit interesting. There's some shoddy looking yellow light, And people will say it's orange, okay, whatever you want to call it. It's yellowish light. I need the lights down pretty far, please. There's red light. Nobody's going to argue with that. And, well, I hope. And somewhere here is green light. So those two lights are overlapping, and I have a knob here that I can turn. You can see in my left hand. And as I turn that knob, I add more green or I add more red and try to balance these two together. So that is greenish, this is reddish for me, and my best match of the two halves is about there. Now, I've been given to understand that this is not right. Does anybody think I'm right? No? You can say no. I hope you don't. I mean, you're all in trouble if you have the same problems I've got. As you will see, I have a mutation that makes my matching abnormal. Can you bring the house light up just a touch? So let's have somebody who's got reasonable vision. You have reasonable vision, but you work here, so I'll spare you. How about one of you? Would you care to turn the knob? Do the match? Come on, it doesn't hurt much. Just turn this. They'll turn the lights back down. Turn this gently until you get the best match you can get between the left and right halves. It won't be perfect because there's no blue light, but is that looking better? Yes? Yes? Okay. So to me, that one on the right looks sort of lawn green. Can you bring the lights back up again? So I want to thank you for coming up here at all by offering you this. You look like a medium. This is a very valuable Rockefeller shirt. It has the motto in Latin, which says, Frigidissima sunt muscae fructorum. Drosophila, fruit flies are way cool. So you get that. Thank you. How about somebody else? Let's see somebody else near enough the aisle. Did anybody else see it as weird? Did anybody not see this as matching? You don't. But come on up, if you dare. Can you get out? I didn't mean to make this hard, but... Okay, so lights back down. Just turn this to your best match. Whoa. Really?
I guess this. I don't know.
Okay, that's cool. So, you guys like that, or? Okay, so generally, okay, so you get a shirt too, but you're normal. It's a very bright color from my perspective. How about somebody else who sees it as abnormal? Does anybody agree with me? Would you come up, please? It's going to be a man, because it turns out that this depends upon how many X chromosomes you've got. Lights down. Yes? So that looks better to me. Got it? Or we'll leave it for now. Okay. Lights back up a second. The only thing more valuable than a Rockefeller t-shirt is a genuine spun aluminum Rockefeller hot and cold drink cup. So, thank you. So you'll see in a few minutes why you and I agree. You and I are right and they're wrong. Anybody else see it? You see it as different. We're looking for people. Okay, one of you back here. This is really real. I mean, we're not faking this. We're mutants. You'll see how in a moment. Lights down a bit. Yeah, either way, just you can try it both directions and see what you like. So what is the general judgment? Is it too red? Too red, okay. So I think it's cool. Lights up, please. Let's see, you probably deserve a drink container, you think? Enjoy. Maybe we'll do one more. But I'm looking for somebody who does not see the majority. Who doesn't see the majority type? So maybe you don't see the majority type? Come on up. So just turn this, you could go both directions and see what feels good. Just start somewhere up here maybe. Is that it? Okay, so you seem to agree with, no? Okay, yes, okay, so you're close to the majority. Lights back up. Let's see, these are large, you're not that large. Maybe you have a drink container, sure. Okay, so you can see there's some real variation. So we found at least a couple of other people in the audience who have, I won't say abnormalities, because I feel that we're better. But anyway, differences from the average person. And I'll tell you a little bit about how this comes about. Let me go back to the slides, please, and I'll turn these down. So as I've mentioned, we have the three different color receptors, the red, the green, and the blue. And each of them absorbs light at a different wavelength. So the shortest wavelengths excite the blue, longest excite the red, and in between is the green. The rhodopsin that I showed you at the first is only active at night in very low light level circumstances. So the gene that's responsible for making the pigment, making the protein that detects these different colors, used to be just a single gene, and I've colored it orange, at least I think it's orange, as sort of a compromise. And 30 million years ago, that gene split into two genes that are side by side on the same chromosome, and it happens to be the X chromosome. Now, what happened at that point actually caused a divergence, it was associated with a divergence in continents. So around the same time, 30 million years ago, Africa and South America, for example, North America and Europe split apart and have been moving apart ever since then, about this far every year. So continental drift is still pulling them apart. So this split in the gene in two did not occur in New World monkeys. So monkeys in South America, with one exception, don't see a difference between red and green. They're the same as dogs and cats. On the other hand, the Old World monkeys, who stayed behind, did have this duplication. So all of the Old World apes, including ourselves, have both a red and a green photoreceptor. Now, as you know, during reproduction, there is so-called crossing over in chromosomes. We each have two copies of chromosomes of each kind, including the X chromosome in a female who's reproducing. There is crossing over in which part of one chromosome is transferred to another chromosome like this, and there's a reciprocal cross in which this part of this chromosome is transferred to that one. And if these genes are in the right order and the crossing over is normal, all that happens is a little information is shuffled. This A gets moved to conjunction with the B prime when it was initially associated with A prime. So this is how there is, in fact, slight variation from individual to individual in each round of reproduction. Because these two genes originated so recently, they still look very much alike, and therefore mistakes get made. And here's the kind of mistake that's made. Sometimes during meiosis, the crossing over occurs not between red and red or green and green, but between red and green, which look very much alike. When that occurs, you can see there are two possibilities. One individual may get a chromosome that has half a red and half a green, that looks like this. The other person may get something that has a whole red, half a green, half a red, and a whole green, like this. Now, if you're a woman, the chances are you will never notice this if you have either of these abnormalities, because you have two X chromosomes, and one of them is likely to be wild type, that is normal. About 5% of people, though, have one of these abnormalities. Now, men only have one of these chromosomes, so 5% of men do have an abnormality. In some cases, a man gets just this chromosome. This chromosome has only one color pigment. The color pigment is a combination of red and green sensitive, but it's like the ancestral one. It's sensitive to orange, if you will. But that person can't distinguish red and green from each other at all. And that's why in our society, when you need to know the difference between red and green, there are other signals. At every traffic light, if the lights are red, green, yellow, whatever it is like that, which one is on top? Red. And if they're side to side, where's the red? It's on the left. So by convention, this allows people who have red-green color blindness not to kill other people when driving. There are other individuals, and I'm one of these, and some of the other people who came up today are of this type. I'm what's called deuteroanomalous. Deutero means second, so the second pigment is anomalous. It's weird. I've got part of a green receptor hooked up with part of a red receptor, and therefore my vision is sort of a compromise. I mix red and green in different proportions. You might mix them, say, 50-50 to get yellow, whereas I and some of the other people will mix it 60-40 or in some other proportion. This third gene does not get expressed, so it turns out, for reasons having to do with genetic expression, just the first two count. You can have, in fact, many more, but they are simply switched off. Now, I'll come back to another bit of genetic information about senses in a few minutes, but now I want to push on to a different type of antenna, and this is a radically different type. This is a fish that's called Aplicylus lineatus, and I just want to show you again how other animals have different sensory lives from our own. This is a minnow that lives in Sri Lanka and lives in southern parts of India. It swims around on the surface of the water. You can see it reflected here. And it eats mosquito larvae, Drosophila, and other things that happen to fall onto the water surface. And the way it does it is very cool. When a bug hits the water, it sends up a ripple. And that ripple propagates over to where the fish happens to be waiting. And when the fish notices the ripple, it will turn around and swim over to the target and eat it. And the interesting thing is, how does it detect that target? Well, it turns out that as ripples propagate, they change in their frequencies. Or more accurately, different frequencies in the ripple travel at different velocities. So this initial perturbation produces some slow waves that move rather slowly towards the fish, and it produces faster waves, higher frequencies, that move towards the fish more quickly. So if you're the fish sitting here and this bunch of ripples comes along, you're going to hear, as it were, from these ripples, something that goes, ew! It starts out high and it comes down. And by how quickly it comes down, you can judge how far away the bug is, right? Because if you're sitting right here, very close by, then it's going to sound like, ew! Just because one frequency rapidly follows the other. And as you go farther and farther away, the ripples spread out more and more, ew! The fish uses that information, and even in total darkness, it swims right to the target and eats the bug. So this really works very well. These animals also have a sensory antenna. On the top of the fish's head, which is just here, this is its nose and its mouth, there are these little ridges that are made out of sort of skin. And in between the ridges, these red things are sensory receptors that respond to vibration. They're called hair cells, and I'll talk about them more in the second lecture today. But each of these has a little mechanical antenna that sticks up and is sensitive to mechanical forces going this way or that way. And there are various of these mixed in together. So when a ripple comes along, it jiggles these little hairs and excites the cells that tell the brain, gee, there's something here. And the neat thing is that each of these receptors, each of the little red things I showed you, has a different orientation of sensitivity. So this particular one here is very sensitive where this red line shows. In other words, it's sensitive to ripples from here and also to ripples from there. This different receptor is broadly responsive. This receptor is sensitive here, here, and so on for all of the others. So there are 9 of these receptors on each side of the body, And the fish uses all this information the same way that we can use, say, to find the source of radio waves by using multiple receivers and homing in on the site from which the waves are emanating. We also use something similar to this in our own hearing, and that comes about when we're trying to localize a sound source. So we're actually quite good at it. Let's see if I've got a quarter or something. If I make a sound, okay, you hear that hit somewhere. You can point to where that hit with a precision of about 1 degree. So out of a full 360 degrees, you have a 1 degree precision based on the sound coming from the source. So, how do you do that? This diagram gives you some idea. A sound that originates, say, over here, to this observer's right, will get to this ear somewhat before it gets to that ear. And you can do some trigonometry if you've got waves moving at a certain velocity and you have a difference in the distance between the wave front reaching this ear and the wave later reaching that ear. You can calculate what the difference in the distance is, delta x, and knowing delta x and the speed of sound, you can calculate how large the time difference is between the two ears, right? So the wave comes zooming along and it goes beep, and then a little later reaches that ear. And it turns out for this 1 degree of precision, we have to have a sensitivity on the order of 10 microseconds. So the ear and the brain are remarkably sensitive to be capable of detecting this very, very small difference. The way it's done in the brain, I'll show you very briefly here. Sound comes into the ear and hits the organ called the cochlea, which again I'll talk about this afternoon. It changes or transduces the sound into an electrical signal. The electrical signal goes into the brain, to this nucleus, the cochlear nucleus, and then there's sort of a race. One nerve fiber carries the information over to this side, and it goes successively to all of these black nerve cells, but it goes bing, 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 up this list. The other nerve fiber stays on the same side and sends information to all of them simultaneously. So if we now imagine a system in which we have sound coming through both sides, you can see that there's sort of a race. The sound coming through this cochlea goes through this nucleus and rapidly goes to all of these nerve cells. The sound coming from this cochlea goes up this nerve fiber and comes first to this, second to this, third to that, and so on through the list. Only one cell, here it's this one, will be simultaneously excited by all of these signals and by this signal as it moves up. So if the sound source is directly ahead, that nerve fiber will light up. If, on the other hand, the sound is to the observer's right, then this signal will get a head start. So this signal will cause all of these to fire. I'm sorry, it's the other way around. This is the right. So this signal will get a head start. It will move farther up before this signal arrives, and this nerve fiber will be the one to be excited. So depending on which cell is excited, our brain knows where the signal was in space in terms of the angle. Let's talk a bit about transduction, and I'll begin talking there about taste and smell, which Vanessa has already introduced to a certain extent. When we taste something or when we smell it, odorant molecules that are in the air or in a foodstuff in our mouth diffuse around, and they diffuse through a mucus layer to finally reach receptor molecules. These receptor molecules, which I've drawn as this bundle of sticks, are proteins. And these proteins have a special site in them into which the odorant or tastant molecule will bind. So this particular molecule diffuses around and then sticks very tightly in a particular place. And when it does that, it triggers a set of chemical reactions. There's something called the G protein. It's just another protein that happens to be stuck there. But it senses when this binds. You can see this pink thing is moved a little bit to the side. That's sent a signal. This molecule turns red. It does something chemical, and it sends a message off, a so-called second messenger, that tells the cell how to react. For example, to set up nerve signals or action potentials. So this is a very common theme, that transduction occurs through the binding of something, and then that triggers a signal which causes downstream effects. Now, the interesting thing about taste and smell is that this binding is very specific, and it's rather like locks and keys. You have different molecules, different proteins, which are called the receptors. And we have in the olfactory system something like 400 of these in humans and something like 1,000 in mice. We have a smaller number of taste receptors. And each of those has a particular molecule or a few molecules that will fit into its particular slot. So you can see each of these odorant molecules, which diffuse around and may bind, has a particular receptor. This key won't fit into any of these other slots. Similarly, the other keys will not fit in anything except the correct slot. This, in fact, again, is subject to mutation. You can have mutations that cause a change, say, in the receptor protein, and as a result of that change, the key will no longer fit. So again, there are differences among us in our sensitivity to various odorants and various tastants. And we'll do a quick test of that. You are equipped with a plastic bag with 4 strips of paper in it. If you don't have 4 strips of paper, you get your money back. And we want the house lights up as far as you can for this one. Now, these papers are slightly different colors, and this will also be a test of color vision because it's very, very hard to see which one is which. But what I want you to do is to take out the white piece of paper first. So the white piece of paper is a control. You don't believe me? You can't tell? Okay, so your science teachers said you guys were smart. So I thought this was the easy part of the quiz. So take out what you think is the white piece, and the idea is do not eat the paper. Eating the paper is bad, but just put the paper on your tongue, moisten it a bit, and taste it. And when you're finished tasting it, take it back out and put the used paper back in the bag. So the what? Okay, quiet again, just so we can hear what we're doing. Shh. So the white paper should taste like white paper. It's a control just so that people don't decide, gee, I've never sucked on a piece of paper before and freak out. So now let's try something that has a taste to it. Find the piece of paper that's a pale green, a pale blue. Okay, do the same thing. Try tasting the pale blue paper. Okay, quiet. We need again to... How many people think it tastes like a dead roach? One... Okay. And how many people does it taste about the same as the first one, just like paper? Okay, so you can see there's real genetic heterogeneity there. So about a third of the people taste something awful, a third taste nothing, the other third don't know how to raise their hands. So you have other problems. So let's try one of the other ones. Try the yellow one, pale yellow. I can... Okay, enough. Okay, quiet. She looks so sick that she gets a shirt. So this tastes really awful to some people. How many people think it tastes terrible? So it's a pretty majority. How many people don't taste anything? Modest number? Okay, there are a few. I'm also a non-taster of that one. And lastly, try the pink piece, the sodium benzoate. Sorry. Okay, so... Okay, quiet again. So this one evokes more different responses. How many people thought it tasted fairly bad? Okay, yes. Why are there more people on this side of the room who thinks it tastes bad? So how many people thought it was a little bit sweet, in fact? I'm one of those. Yeah. And how many people thought neither of the above, something in between sweet and bad? Yeah. So it's, about a third each. So All of this comes from differences in the shapes of your receptors. I mean, different ones among us have different locks into which these keys fit. And depending on which particular ones you have, you evoke an entirely different sensation. Now, the last part of this test, there are two more parts. One is to put all of the pieces of paper back into the bag, because we might want to use them again next year. And you'll later be asked to throw them out as you leave the room so that we don't have to pick them all up. Now, the same sort of transduction occurs in the case of light. I remember mentioning rhodopsin, which is our protein for sensitivity to low light levels. It looks just like these other receptors, and it's also bound to a G protein. The only difference with rhodopsin is, instead of having a molecule that drops in from something you taste or smell, it has a molecule that's glued in place. It has a molecule called retinal that's stuck there. And the retinal molecule, when it captures a quantum of light, changes its shape from this to this. That then triggers this G protein, and it goes off and sets up the response in the rest of the cell. Here's, in fact, a picture of the retinal molecule. It starts out stressed, bent in this particular position. When it captures light, it flips to this other position, and that then is the trigger that excites the rest of the response. Let's turn next to amplification. And there are two types of amplification I want to emphasize. One of them occurs in this really simple animal. This is a paramecium. And a paramecium, as you know, is a single cell creature. It has a lot of cilia on its surface. I got tired of drawing thousands of cilia, so this one is naked. But with its cilia, it can swim. So when it's just out having a good time, it swims in a sort of a sinusoidal pattern. In fact, it spirals like an American football when thrown. And as you know, when you spiral a football, it tends to be stable. It tends to go straight. If it's tumbling end over end or something, it goes in a crooked manner. And the paramecium does the same thing. The paramecium has two responses in life. One of them is that it sometimes runs into a wall, boink. This is a somewhat impoverished existence. When it runs into a wall, it does the right thing. It backs up, and then it goes off in another more or less randomly chosen direction. And I'll show you in a second how this comes about. The other thing it does is run for its life. If something bigger comes along and tries to eat it, it goes faster. And interestingly, when it goes faster, it doesn't just go faster, but like a poorly thrown pass, it wobbles. And of course, the wobbling is helpful, because as it wobbles, it makes it harder and harder for this, which is a zebrafish, to catch up with it and grab it. So how does it do it? And it does it with a small number of ion channels. Ion channels, Vanessa mentioned during the introduction, are proteins that allow ions to flow across the membrane. These animals have a bunch of calcium channels at their front end, which is here, and I've showed them sort of shaded in green. I think it's green. And it has another set of mechanically activated potassium channels at its other end that I've shown in sort of an orange. So if it gets bonked in front, these channels go off. If it gets bonked, say, by a predator from behind, these channels go off, and they elicit two different responses. In this case, if it gets hit in the front, the response is to back up and swim in a new direction, because you've hit a wall, dummy. If these get hit, then it speeds up, and it ceases to go like an American football. It no longer spirals. It just goes as fast as it can, and it tends to go erratically, and thus to avoid the predator. So quick review of ion channels. Excuse me. First of all, ions, remember, are atoms that have lost or gained electrons. So a cation has lost one or more electrons. It has a positive charge due to its nucleus. And this includes sodium, potassium, and calcium ions. And an anion is similar, except it's gained an extra electron. So something like chlorine or chloride is an anion. I'm not sure it's possible. You'd have to be really swift to bonk one in the middle. I expect what would happen is the two responses would cancel out. I mean, it will be some overlap between the two, and the animal will be annoyed, understandably. But that's it. So the way these ions get across the membrane is through channels. So every cell in the body, as you know, and bacteria and whatnot, all have a membrane around them that's made up of lipids. And into this membrane, you can insert an ion channel. It's made of protein. It has a number of cylinders or the like that pass across the membrane. But importantly, it has a hole, a pore through it. And typically, it has a gate that can be opened or closed. So the channel can be active with the gate open, it can be inactive with the gate closed. So the way in which signaling typically occurs is that ions are built up on one side of the membrane. This is done by using cellular energy sources such as ATP and what are called ion pumps. And once this charge is built up on one side, it's just like a battery. It is, in fact, a battery. There's energy stored here. And this energy can be released by opening the channel. So when the channel's gate swings open, now ions can rush out and send some sort of signal. So, for example, if these ions move from here to here, this compartment will become less positive, this compartment will become more positive, and that's what happens when an action potential propagates along a nerve, as I mentioned at the very beginning of the talk. And there are various ways you can open a channel. You can open it by membrane voltage. That's what happens in an action potential. You can open it with a chemical neurotransmitter. That's what happens at a synapse when one neuron talks to another. And you can open it with mechanical stimulation. That's what happens in the case of the paramecium. The channel is directly opened by being bonked. So here is, in fact, the same paramecium running into the wall. An ion channel in this region is going to be stressed by membrane stretch, and now current will go through that. All these numbers you don't need to worry about, but I just want to show you one can do honest bookkeeping and ask how much energy goes into this system. In other words, how extensive is the bonk? How much mechanical energy does it produce? And there's a certain amount. How much energy occurs as a result of the ions flowing across the channel and setting up an electrical response in the cell? And you can do the accounting for that. And the important thing is the difference or the ratio between the two is 2 million times. There's an amplification of 2 million times because one bonk affecting one channel can allow millions or anyway hundreds of thousands of ions to flow across, and that gives a huge amplification. So this is one of the ways in which you can amplify a weak signal like a tiny bonk into a really strong signal that causes the animal to get up and go. The other kind of amplification is chemical. And again, you don't want to know what's going on here, except that this is what happens downstream of the photoreceptor events I showed you before. Remember, rhodopsin captures light, it changes its shape, and when it does so, it interacts with this G protein called GT. That then sets off a chemical cascade. This changes its configuration, it interacts with something else, phosphodiesterase, that interacts with something else, cyclic GMP, that interacts with the channel, that allows sodium and calcium ions to flow across. So all these things are triggered like a series of dominoes downstream of the initial event. But the important point of this is, again, amplification. One rhodopsin can trigger 1,000 of these reactions. One of these reactions can trigger 1,000 of those reactions. And again, opening one channel can affect the flow of 600,000 ions. So when you multiply all of these things together, you get about an amplification of about 500 billion. In other words, this is, 600 billion times amplification from the energy of one photon of light going in to the final response at this other end. So again, we're sensitive, in fact, to single photons of light. In the very darkest circumstances, we can actually detect one quantum of light, and we are able to detect that in our brain because of this enormous amplification that finally produces a big signal that the brain can't ignore. Going back to our earlier diagram, we can also see that this cascade of chemical reactions is useful for adaptation. So in this case, after this amplification step, owing to the chemistry, there can be a negative feedback that turns down the sensitivity. In other words, there are additional chemicals that reduce this gain, and so on, and make us less sensitive. And this is exactly what we experience when we go out into a bright light. I mean, you're sitting in darkness now, your visual system has tuned itself up so that all of these activities are pretty strong. And they would be even stronger if you were in a totally dark movie theater. But when you step outside, you don't need that amplification anymore. Adaptation will occur, it will turn down the sensitivity, so you'll be blinded for a few seconds by the bright light, but then you'll adapt to it and you'll be seeing normally again. And similarly, when you go back into a dark environment, it takes a few minutes, up to 20 minutes, to totally turn all of these gains back up in order to get the maximum gain that's possible. The very last point is this idea of encoding, that is representing the stimulus as a pattern of nerve impulses. Because remember, all the brain finally gets from all these nice sensory receptors is a pattern of spikes, pop, 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 pop. Here is a typical set of responses in which we have a stimulus of any sort. So this could be stretch, it could be light, it could be sound. We'll use 1 unit of stimulus and we'll get a certain response, pop, 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 pop. If we use 10 units, not 2, but 10 units, we'll get roughly twice the response, pop, 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 pop. If we use 100 units, we'll get approximately 3 times the response, brrr up. So notice the ratios. This is 1, 2, 3 units of response, but 1, 10, 100 units of stimulus. So if we make a graph of this, here's a graph of the stimulus in linear terms and a graph of the response in linear terms. You get a curve that looks like this. It rises quickly at the beginning. That is, you get a strong response to a weak stimulus. But as the stimulus gets stronger and stronger, the response rises less and less. There's sort of a saturation. And as many of you know, you can also see this by taking logarithms. If you take the logarithm of this is in fact, 0. The logarithm of this is 1. The logarithm of this is 2. So the response in terms of the log of the stimulus is a straight line. And this is a very common thing in our lives, and it makes good sense. We want to be very sensitive to weak stimuli because, again, this enables us to hear the predator before it eats us. It allows us to hear the prey before it can escape from us. We want to make the system very sensitive at low levels. But as it gets louder and louder, or brighter and brighter, or the knee gets hit harder and harder, you care less and less about how big the response is. So you get saturation, or you get a weaker response to progressively bigger and bigger stimuli. And in fact, there's a general relationship for this. It's called, after the people who named it, the Weber-Fechner relationship. And it says what's shown here, that the perceived difference in a response for a stimulus change delta S is proportional to the magnitude of the stimulus you've already got. So in other words, the change in the response to a change in stimulus delta S is delta S divided by the pre-existing stimulus. So what this means is if you have a large background stimulus, let's say you're in a very bright light, you need a large increase in light in order to get a response that you can detect. But if the background light is very low, if this S is very small, then a very tiny change in light will give you a perceptible response. You can also change this into a logarithmic relationship that says the response is proportional to the logarithm of the stimulus divided by some threshold level of stimulus. And as I said, this is characteristic of a large number of our sensory receptors. So we are doing okay in terms of time. I'm going to end the talk at this point, but we'll then take some additional questions from the audience. Thank you.
Thank you very much. I'm wondering if you could comment a little bit about molecules that we're ingesting, for want of a better term, that we don't really perceive, yet they cause a response, particularly pheromonal or smells or tastes that we don't understand, but we get some kind of response from it.
Right, so do you want to comment about pheromones? You're more pheromonal than I am.
Right,
Is this live? Yeah.
between species, there's a lot of communication that, guides their behavior that they may not actually be, externally aware of, that it's part of their, underlying modulation of behavior. It's very common in other species, and actually one of the demonstrations that we'll be showing today is actually how fruit flies communicate with each other through chemicals that they emit and that are perceived by other fruit flies and interpreted to say, are you the right sex, are you the right species? It is something that actually has been speculated. If you ever look, there's many, cases where people actually would like to identify pheromonal chemicals that would be in part of guiding human behavior. I think there's a whole, drug industry sort of dedicated to that. But I think there's actually very little evidence. There's only one, I think, sort of example of pheromonal communication that's thought to occur, which is in humans, and that's the sort of the synchronization of female menstrual cycles. So that there probably is, are some chemical cues that we are emitting off that we don't even know, and that other, other humans that we're cohabitating with are probably picking up.
If perfume manufacturers could find such a pheromone, obviously it's worth a lot of money. So people are looking hard. Other questions?
Why? You had the molecule that said it was set up in your slide. Why was it that double bond that flipped or rotated? Why not some of the others?
Yeah, so the question is, in rhodopsin, there's a particular strained double bond that is the 11-cis bond, and it's the one that rotates when the system captures light. And it's because it is molecularly strained. It's metastable, which is to say that it wants to relax to this less strained configuration. Interestingly, there's a problem that photoreceptors have when you think about it. You've got all these molecules of rhodopsin. They're all strained. They all want to be released. And in fact, now and then, just heat energy, thermal energy, Brownian motion, will knock one from the strained state to the relaxed state. And that will, of course, be seen as a quantum of light. So if just plain heat knocks it from one state to the other, our brains can't tell that from a real quantum of light. So the system, through evolution, has had to deal with this problem. It wants to be sensitive enough that it will reliably respond to a single photon. But at the same time, it doesn't want to respond all the time just to heat energy, which would make us see just a whiteout. It turns out there's a so-called dark light. That is, that if we go into the darkest circumstances, we will be constantly bombarded by a certain number of these molecules undergoing a spontaneous switch. And that sets a limit to how few photons we can see. I've said that we were sensitive to single photons. That was what scientists call a lie. The reality is, if we catch single photons, each photoreceptor can measure a single one, but we have to capture about 5 simultaneously in 5 different cells in order to finally say, I'm sure I saw one.
So would it be fair to say that all the senses work similarly because they're all stimulated by something which triggers a feedback mechanism, which would trigger our response?
Yeah, so that's exactly the idea. You hit the nail on the head. I mean, evolution is looking for solutions to problems. And the problems are the same for all the senses. How do you detect the weakest sense of that kind that you possibly could? Because as I said, there's a big selective advantage for that. How do you categorize the particular sensation? You want to be able to tell the difference between a dead roach and a delicious souffle so that you can enjoy one and abhor the other. You want to be able to adapt so that if something gets too loud, too strong, too bright, whatever, you can turn it down and attend less and less to it. All of those challenges are faced by all of the senses. And what I was trying to get across, in fact, is there's sort of generic solutions to them. Again and again, you use antennas to capture the energy. You use tricks to transduce the stimuli. You use amplification by cascades of proteins or by ions. And finally, you encode everything the same way. This is something that was realized as long ago as the 1860s, that if you record from a nerve somewhere in the body, that nerve will give similar signals, whether it's recording, say, the response to being stuck with a pin, ouch, or it's responding to heat, or it's responding to cold, whatever it happens to be. All the signals look the same. The signals from my eye that signal light, the signals from my ear that signal sound, those signals are identical. So it's rather like what would happen if you tapped into a phone line, as the NSA does, outside the university. When you tap into that line, what are you going to see? You're going to see ones and zeros. But the ones and zeros look the same, whether it's bank account information or, tweets, whatever it happens to be. They're all ones and zeros. The senses work the same way.
Is it possible for encoding to take place at some point other than at the end of a stimulation cascade? Or would it be turned off?
No, usually, I guess it's a question of definition. The information is usually carried from a sensory organ into the brain by a nerve fiber. And by definition, what goes down that nerve fiber is the neural code. It is, in fact, a representation as ones and zeros or as action potentials of whatever has happened before. So encoding is always occurring at the step where the information goes from the sensory receptor to that nerve fiber and then on into the brain. And of course, the brain then needs to decode it. We're not talking about that at all today, but there's a reciprocal problem. Once the information gets into the brain, it has to know where that nerve fiber came from. And that's the other half of what was discovered in the 1860s. Johannes Müller is the one who realized this. Because those signals all look alike, their significance in the brain depends on where they're plugged in, right? So it's important that a signal from the eye go to the right part of the brain to carry visual information. it's important that stimuli from the ear go to the right part of the brain to carry auditory information. And in fact, people like Muruganka Sur at MIT have done experiments in which they've swapped those wires in developing animals. And lo and behold, the signals get through okay, and they're even being interpreted by the wrong parts of the brain. So you have visual parts of the brain interpreting sound, auditory parts of the brain interpreting vision. So the brain is plastic or flexible enough to accommodate such misconnections.
I'm extremely interested in photon transduction and sensory information, as you describe it, because we also use photons in the skin for the purpose of metabolizing for vitamin D. And in terms of sensory function, it seems to me that it's not just sensory information that we're extracting from the environment, but actually regulatory information that's a part of human regulatory metabolic functions that are tuned into the environment. So I would just like to add that as a thought, because
sure.
When we talk of stimuli, we're sometimes forgetting that we're actually being regulated by the environment also in order to function better within it. Your thoughts on that? Any?
No, just the only other comment that's related to that is I showed you that the visual response in rhodopsin is very closely related to the taste and smell response. Probably there was an ancestral receptor, I would suppose it was a taste or smell receptor, that allowed organisms to detect something in their environment. And one day in a mutation, it became possible for one of the things that fit in there, rhodopsin, to become stuck. And now you've got a light receptor. So this is, again, the way evolution tends to work. It takes whatever is at hand and manipulates it to do something else. And as you say, I think there are probably other functions being served as well. Yet another function of this sort, which we're not very aware of, is there are other proteins called cryptochromes, which capture light. And these are what are responsible for our circadian rhythms, our sense of light and darkness. And they give us such benefits as jet lag.
Is that what synesthesia is? So, like, if the wires were changed, you would experience, like,
Yeah, that's a really good question. So what she's asking about is synesthesia. So syn means with. Synesthesia is a sort of fusion of the sensation owing to different kinds of stimuli. And some people have this. I'm sure some people in the room have it. There are people who, for example, when they see numbers, see them in colors. They have certain colors associated with 1 or 2 or 3. So they have sort of, within vision, a synesthesia of what is just a plain black and white numeral with color vision that somehow invades that sensation. And then more extreme, there are people who, when they hear a certain sound, will have a particular feeling in their body, or when they see a particular thing, will hear a particular sound or whatever. We don't know where this comes about. It might well, as you suggest, come about from the fact that there are changes in the wiring in the central nervous system. So information that's ordinarily routed into the visual stream is somehow bleeding into the auditory stream or vice versa. We don't yet know how to see that. Perhaps with magnetic resonance imaging or the like, one would be able to pick up signals that show where and how that occurs.
Could you speak a little bit to, for example, the sense of smell and its connection to memory, and how olfactory senses can trigger things, for example, images?
That's a tough one. Can you do that?
Yeah, actually, I think one of the most amazing things about the your sense of smell, if you think about it, is just the complete complexity of chemicals in our environment. And the thing I like to think about is that if you were to take, for example, smell a glass of orange juice, you would tell me right away that it was, you close your eyes, you smell, it smells like an orange, right? So it's not that you can actually say what chemicals it smells like. You can only say what the object that you associate it with. And that's because actually olfaction really relies very heavily on taking, on basically learning and memory as a way to make sense of this incredibly complex set of chemicals that exist in the environment. We encounter them in many, even natural odors have like more than 200 or 300 different chemical molecules within them that our brains actually detect each of one of them by the receptors that Jim spoke about. And then it activates multiple different pathways that ultimately are integrated in some higher brain center and that are stored there as a particular sensory object, the smell of an orange. So there is this really heavy reliance on learning and memory to make sense of the chemicals in the environment, and that means that, they're very evocative. They really sort of take you back. You smell something, you smell, an object, you smell a perfume, you smell something, and it reminds you exactly of the time and the place that you probably may have first encountered it.
The other way of looking at that problem is to sort of invert it and think about cooking. It takes a very special person to deconstruct
That's right.
an odor or smell. So if somebody gives you a nice souffle or whatever it is, and you say, gee, what is in that? An ordinary person is clueless. It takes a person who's a great cook, a great chef, to be able to say, gee, this is a certain amount of almond and a certain amount of lemon and a certain amount of whatever to make this up. You've got to learn that.
I have two questions. First question is, as we age, other than regular average deterioration of the body, does smell and taste also, we lose some of it? And my other question is regarding the taste, what are the benefits from evolutionary point of view to having us in this room, for example, being able to taste completely opposite tastes of the same material? So for some of us, just the three of us, two of us, the blue strip was similar to the white, but for one of us, it was completely bitter. So what was the benefit of it was it the region where we developed or anything else?
So can I have this pad turned on for a second? We have in our olfactory, well, I'll turn first to taste, which I should probably turn over to Vanessa, but in the taste bud, we have a series of responsive cells. We have a series of responsive cells, and some of them have receptors on their surfaces that allow them to be sensitive to specific things such as sugar. And so we have sweet receptors that respond more or less only to sugars, and they send information along a nerve fiber into the brain, no problem. We have others that are sensitive to salt, sensitive to sour, sensitive to something I'm forgetting. Okay, but the one that's most complicated is bitter. There are a lot of different chemical receptors, that is protein molecules, all of which signal bitterness. And most of what I was demonstrating was that. So this cell, which happens to be a bitter sensitive cell, may have some little black circle receptors. It also happens to have some little red triangle receptors, and it might even have a little purple ellipse receptor, and in fact up to 30 others. So a single bitter receptor cell can have dozens of different types of bitter receptor molecules on it. And any of those will provoke a sense of bitterness. So that's why we have the mutations and the differences among us that I showed you today. Some of us might lack the red triangles. Some of us might lack the purple lozenge or what have you. We will all have many other bitter receptors. The reason that bitter receptors are lumped together is that, of course, sugar is a good thing to find if you're looking for energy. Sour and other things of that sort may also be useful in finding things that are good to eat. But bitter usually signals poisons. A lot of plants produce so-called alkaloids, which are bitter-tasting substances. They produce them, in fact, as poisons to keep insects from eating them and also to keep us from eating them. And in fact, there's a correlation with our receptors and what we like and don't like to eat. So the family of drugs or family of compounds similar to those that you were tasting includes substances that are found in broccoli. And as we know, how many here hate broccoli? Right? And you may have tasted those substances. People who dislike broccoli, Brussels sprouts, cauliflower, broccoli rabe, et cetera, tend to have a particular group of these receptors. Other people who think broccoli is, sweet as honey, lack those particular receptors.
Yeah, I'll just add one sort of extension of this, which I think is really fascinating, is that there is some interesting evolutionary variation in taste perception amongst different animals. For example, obligate carnivores, they wouldn't need necessarily to taste sugar anymore. And so many of them, including polar bears, for example, have lost the receptors that were responsive to sugars have been lost. And now they actually have extra receptors that taste amino acids and other chemical compounds that are found in meat.
Do cats have sugar sensitivity? I thought cats were low in sugar.
I think cats are another, they're also low in sugar sensitivity. So if you feed your cat sweet food, they're eating it for some other reason.
Because they're starving, so. Yes. Sorry, there was another half of the question, remind me?
As we get older.
Oh yeah, so the other thing about these receptors, and it's also true of the olfactory receptors are actually a kind of nerve cell that have a sort of a tentacle sticking out, And there are tiny cilia which have the receptors on them, and then they have an axon that goes into the brain. These cells are constantly dying and turning over. There are new cells down here at the base of the epithelium that undergo mitosis. Those are mitosis, and those then reproduce these cells. But as we age, we gradually deplete these cells. We have fewer and fewer capable of reproducing, so as we lose these cells, we become less and less able to replace them. There are also some chemicals that clearly kill these things off. So tin chloride, stannous chloride, really will wipe them out. And a lot of things in the environment, particularly in laboratory environment of the sort we're in, damage these, and so we tend to lose our sense of smell. Many people who work in chemical environments and the like have lost theirs altogether.
Yeah, I'm curious to know the response to bright and low light conditions. Why does it take 20 minutes for the amplification for sight to return? In other words, to see very low level light, whereas when you go to bright light, it's almost instantaneous, or it's very quick that you have that adaptive process.
Yeah, it's simply a question of the biochemistry involved. It turns out that a lot of the reason that there is adaptation is, I showed you the photoreceptor molecule sitting in a protein interacting with the G protein and setting up a set of signals. It turns out that one of the ways in which on bright light the system is desensitized is that phosphate groups are added to the rhodopsin. So they're actually new molecules, stuck onto the rhodopsin, and until those have been peeled off, it's not going to be sensitive again. The other thing that happens in very bright light is that a good fraction of the rhodopsins, remember the rhodopsin has the ring to it, I won't draw all the double bonds, it has an ionone, yammer, but when it's in the tense state, it's like that, and then it relaxes to the straight configuration under light. It's got to be cocked again. It has to be restored to its original state. And the way that's done is rather complicated. The molecule actually falls out. It goes into the pigment epithelium, which is a black layer of cells behind the eye. It gets chemically isomerized there. It diffuses back. So all of this toing and froing is what takes the 20 minutes.
I think we should just thank Jim once again for really a fascinating morning.
The second talk is going to be more specialized. It will deal with just one sense, the sense of hearing, which happens to be the sense that our research group works on. And by way of an introduction, I should tell you some of the reasons that motivate us to study this. One of them, obviously, is a lot of people have trouble with hearing. In this society, in this country, about 30 million people, which is 1/10 of the population, have significant hearing losses. Some of you probably have hearing problems. You certainly know friends or family who have hearing problems. So it's a very broad societal problem, and it's one that gets worse in an industrial society with more noise in the environment and now more noise from entertainment devices and whatnot. So we would like to understand something about where these problems come from and what to do about them. Now, hearing loss stems from a variety of different conditions. First of all, there are more than 200 genetic conditions that cause hearing problems, some of them in isolation or sometimes in association with blindness or problems with the kidneys or other organs. Next, there are various infections, such as maternal rubella or later in life meningitis, that can damage the cells of the inner ear and deafen one. Next, there are loud sounds, what's called acoustic trauma. And this can be loud sounds in an industrial place, it can be loud sounds in the military, it can just be stereo or ear pods turned too far up. There's something called presbyacusis, the loss of hearing with age, and that's very widespread in the society. By age 65, about 1/4 of us have hearing problems, and by age 80, it's 50%. So this is extremely widespread and becoming more and more prevalent as the society ages. And the other cause is simply drugs of various sorts, not illicit drugs, but there are various legitimate drugs that have to be used by physicians sometime that have as their major side effect damage to the inner ear. So there are drugs that are used for severe heart infections, bacterial endocarditis. They can damage the internal ear. There's the major chemotherapeutic agent for ovarian cancer called cisplatin, and it also has to be used in life-threatening conditions if a woman has ovarian cancer but it can cause partial or complete deafness as a result. So all of these are issues that lead to the prevalence of human problems with hearing and things that we'd like to deal with. The impact of the hearing loss is substantial. As you can imagine, young kids who are born deaf for genetic reasons have a real problem with acquiring speech, and therefore all sorts of symbolic communication. And as a consequence of that, there's been a real effort in hospitals to test every child when he or she is born with a very simple test, and I'll show you how it works a little later, that can tell whether the ear is normal or not before the child ever goes home. And that way, kids who have a hearing problem get detected at a few days of age, and then one can do the appropriate thing to help them. Perhaps surgery will help in some cases. Perhaps they will simply get a cochlear implant, which I'll talk about at the end of my lecture, or they may learn American Sign Language. But any of those ways, they begin to communicate at a normal early age, and this, of course, greatly improves the development of their thinking process, of their cognitive abilities. For older people in the society, and many of you will see this in your own family, hearing loss deprives people of contact with their family and their friends in their workplace. A lot of older people feel isolated. They can become suicidal as a consequence of simply not feeling plugged in any longer. And for people of the intermediate years of life, which includes you, the problems can also be very severe. Any of us can wake up deaf tomorrow, particularly from bacterial infections. And people who are suddenly deprived of hearing find themselves incredibly depressed and isolated, and it leads to depression and leads to suicide. Helen Keller, who was, of course, blind and deaf, said that blindness deprives us of our contact with things, but deafness deprives us of our contact with people. And even though most of the people around you are jerks, you will find that the sort of casual interactions you had over in the other building when you were chatting with people on lunch and so on, those are very important in sort of setting you in your social milieu. And if you're deprived of that, all of a sudden you feel very, very isolated. The other thing hearing does that we don't pay any attention to is it's sort of a distant early warning system. You know if you cross the street that you can hear a fire engine or a police car coming with a siren. And if you're in a building and it catches fire, you can hear the fire alarm and whatnot. You take that for granted. But in fact, if you haven't any hearing, you have a sense of vulnerability that hearing people really can't ordinarily articulate. At any time, you may feel something bad is happening, you don't know what it is. And people without hearing also can be startled. If you just have somebody come up behind you and put their hand on your shoulder, and you're not expecting it because you don't hear them, it's quite frightening and disturbing. So we want to deal with these problems, we would like in the long run, to remediate hearing loss by restoring the cells that have been damaged. And I'll tell you about those cells. The other reason we're interested in this is from the basic research perspective. And that's because hearing has these really remarkable properties. One of them is we can hear frequencies as great as 20 kilohertz. That's 20,000 cycles per second. Bats and whales can go to 100 kilohertz. Now, just by comparison, if we consider, say, vision, you know perfectly well if you see images 24 times a second or 30 times a second, as on TV and motion pictures, it looks like a continuous image, right? Your eye is fooled by the fact that even changing the picture 20 times a second is fast enough that the eye can't detect it. Yet the ear is somehow working at 20,000 cycles. It's working 1,000 times faster than those other senses. So we'd like to understand how that happens. A second remarkable feature is just how sensitive it is. At the threshold, the very faintest sound you can hear vibrates the inner ear's parts by about 0.3 of 1 nanometer. So a nanometer is a billionth of a meter, that much, and it's 0.3 of one of those. So it's about the size of a reasonable molecule. It's not, the size of a cell, let alone an animal. It's this teeny tiny vibration. So how can the thing made out of proteins and lipids and this and that, how can such a thing be reliably sensitive to an atomic level of vibration? And the third remarkable property is what's called dynamic range. From the very faintest sound you can hear, which is defined as about 0 decibels, to the loudest sound you can tolerate before your ear is damaged, which is about 120 decibels. That's 6 orders of magnitude, a million-fold range in the size, the amplitude of sound that we can tolerate. Or if you view it in terms of power, it's the square of that. It's a trillion-fold, 10 to the 12th range in the power of the sound from the faintest to the loudest, which is remarkable. There's no human-made device, no meter, no dial, no nothing that can cover 12 orders of magnitude. So we again would like to understand how this comes about. So what I'm going to do is to tell you about the cells that do this and how they work, and then at the end, what might be done to replace them when they're damaged. I'll begin just by placing all this in the context of what your ears are doing right now. So right now, as you listen to me, you're listening to signals that are somewhat like this. If you simply recorded what's coming out of this microphone, you would get an electrical signal like this, with a series of pulses representing the different syllables. And down below is the same information, except it's been turned into a sonograph. And a sonograph is a kind of graph of about 2.5 seconds of information, but now it's portrayed in terms of frequency. So there's some syllables like this one that have a very high frequency content. There's some that are intermediate, and then a lot of them that are relatively low frequencies. And this is in fact my voice, and what this piece is the end of a poem. It's called Fern Hill by Dylan Thomas, whose 100th birthday is this year. As I was young and easy, in the mercy of his means, time held me green and dying, though I sang in my chains like the sea. So this is though I sang in my chains like the sea. And you can see that these low frequencies are the vowel sounds. Though, ang, ains, ma. I'm from Texas, so this here is a ma. The consonants are high frequencies. Sang, chains, see, and whatnot. And this is what your ears are doing all the time. You're going to hear in this talk 53,000 different speech sounds, if I finish it. And to understand it all and to pass the quiz, you've got to understand them all. And yet you do this without ever thinking about it. You're doing this on the fly in real time. You're picking out these combinations of consonants and vowels and looking at the different frequencies and whatnot, and using that to decide in your brain what is being said and then what, if anything, what it means. So how does this work? Let me show you, first of all, how sensitive it is and how our sensitivity to different things varies. I'm going to play some sounds that change in magnitude or amplitude. So I'll play a tone, and then it will get 200% louder. That is, it will be 3 times as loud. And you can very easily hear that. Okay, yeah, a lot of you will be deaf at the end of this, I should say. Now 100% increase in amplitude. That's easy. 50% change. 20% change. So that starts getting to be a little hard, right? Even though it's changing 20%, which for a lot of things is a lot, you can just barely tell. 10% you mostly can't tell. Right? So we're not terribly sensitive to the loudness of sound. And I want to contrast that with our sensitivity to frequency. So remember, we could hear down to about 20% change. We'll now look at frequency. And in frequency, I'll start with a much smaller change, just 2%. Piece of cake, right? 1%? 0.5%? So those are all quite easy to pick out, much, much smaller than the differences in intensity. 0.2%, 0.2% gets to be a bit of a challenge. So those of you who play stringed instruments can hear it, the rest of us can't. And 0.1% no human can hear. Well, I said no human. I'm not talking about nerds. So a semitone, a single step on the piano, is a change of 6%. So 6% is 3 times this much. So we can hear something that's only 1/30th of the difference between a black and the white key on the piano. So we're very, very sensitive to frequency, not so sensitive to amplitude. And when you think about it, that makes sense. Again, if you're out in the wild, if you're looking for dinner, a cheeseburger, a gazelle, whatever you're looking for, you want to be able to tell differences in frequency, because differences in frequency tell you about what an animal is. Different animals have different pitches of voice, just as we do. You want to tell cries from each other. You don't care if they're this far away or that far away or whatever. The distance isn't so important, and therefore the loudness isn't so important. But frequency is really key. And it's the same in our own hearing. Our capacity to understand speech depends critically on having really sharp frequency discrimination. But we can understand the same thing whether it's very loud or very quiet. We have no trouble accommodating to that. So here's where the action occurs. This is the cochlea, which is this coiled, elegant, snail-like device. The name cochlos is Greek for snail, for obvious reasons. It's about 8 millimeters across, which is the size of a chickpea, a garbanzo bean. And it's part of the labyrinth within the temporal bone of the skull, right here behind the ear. And I've mentioned the other organs in the labyrinth this morning. There are 3 organs that are responsible for our ability to carry out balletic motions. These are the semicircular canals. So these have liquid inside them, and when you spin around, the liquid spins around, and that gets detected and sent to the brain. There are 2 others, the utricle here, the saccule here, that are sensitive to linear acceleration. So this is the saccule. It's sensitive to up and down motions, such as making us seasick or airsick, as well as telling us where gravity lives. The utricle is what's involved in side-to-side motion. So if I move like this, my ability to tell where I am comes from measuring signals in the utricle. So if we consider the cochlea, we can imagine taking this structure and unrolling it. So this would then be a bony tube about 30 millimeters in length. That's 1.5 inches. It's filled with liquid, and down the middle is this elastic structure that's called the cochlear partition, or we can call it the basilar membrane. This is hooked to the little bones of the middle ear, and you've already learned that there's an eardrum attached to the malleus, or hammer, the incus, or anvil, and the stapes, or stirrup. So these are the 3 smallest bones of the body. And when sound comes along, it pings the eardrum, and the information is then passed through those little bones. So sound, as you know, consists of alternating compressions and rarefactions of the air. The compression pushes in on the eardrum. That moves the 3 little bones to your right, increasing the pressure in this upper compartment and pushing the membrane down. When instead there's a rarefaction, that sucks the eardrum outward, everything moves to the left, this gets moved up. If this structure were homogeneous, if it were just like a guitar string, then the whole thing would just oscillate up and down, much like a guitar string, and move in a very simple way. But in fact, there's something much more complicated that occurs. This is not a guitar string, it's not homogeneous. One end of it is very broad and floppy, and it vibrates at low frequencies. The other end is very taut and narrow, and it vibrates at high frequencies. And in between, there are various other frequencies represented. So the cool thing about it is, if you listen to a complex sound, I've showed you that each time I make a sound, chains, C, whatever, there are multiple sounds being produced. Those sounds are broken down in real time. The high frequencies cause a vibration here, the low frequencies cause a vibration here, and the intervening frequencies are at various positions in between. So this is what's called a frequency analyzer. Those of you who know the math know about the Fourier transform. This is a real-time Fourier transformer. It breaks down whatever kind of signal you put in into a series of sine waves, and each of those sine waves gets distributed to a different position along the length of the cochlea. We know this by measuring with interferometry. We can open the ear of an animal, such as a mouse, or we use chinchillas, make a small hole in the bone of the ear, and shoot a laser beam in, bounce it off this elastic basilar membrane and back into the laser, and then we can carry out interference. We compare the wave going in and the wave coming back, and we can infer how much vibration is occurring at this point. So when we do real measurements, we see something that looks like this. This is a little less than 1 millimeter along the length of a chinchilla's cochlea. And along the basilar membrane, you see this beautiful wave that comes from the left and travels or advances from left to right, peaking about there and then beginning to fade away. Now this is a traveling wave. It isn't a wave that's standing still of the sort you see with a guitar string or something of that stripe. And with any sort of luck and my handy rope, I'll just point out the difference. Looks like my handy rope has gotten snarled, but it keeps doing that. But anyway, ouch. So if you have a guitar string, it moves with a single mode, up, down, up, down, up, down, like that. The difference with a traveling wave is that it travels. So depending on the frequency, it does that, right? You see the wave propagating along the rope. So high frequencies propagate a short distance. They're here at the base of the cochlea, and they go only as far as the nearest photographer, and then they stop. And if you have a somewhat higher frequency, somewhat lower frequency, it goes farther. And the very lowest frequencies will go all the way down to the second, or maybe it's the same photographer, at the far end. So this is, again, how the sine waves get broken down. A real complex sound will have all this happening at once. There'll be some high frequencies, there'll be some low frequencies. Each of them will cause a wave that peaks at a different discrete position. And all this is happening in real time. So as fast as the waves can occur, your ear is breaking the sound down and reporting it to the brain. So the consequence is this. Along the length of the cochlea, there are some 16,000 cells that I've shown in yellow. They're called hair cells. They're not the kind of hair that I'm losing, but you'll see in a minute why they are called hair cells, because they have miniature hairs on top of them. Those at the base are sensitive to high frequencies, those at the apex to low frequencies, and again, all the other sounds in between are represented. Those cells are then connected to about 25,000 nerve fibers, which I've shown in blue, which carry information into the brain. So again, when a particular tone is sounded, say middle C, just these hair cells are stimulated, and the nerve fibers connected to them will fire action potentials. Again, brrr, the cells that are nearby here on the two sides will fire somewhat less, pop, 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 and I've shown that in yellow and orange. So let's look at a real sound. Here is the word chains from the piece of speech I showed you before. So it begins with the sound cha, a loud, high-frequency consonant, and that's going to cause stimulation of these two bands of cells at high frequencies near the base of the cochlea. Then the ains part of it is going to stimulate here and here at two relatively low frequencies, and it's somewhat weaker. And then finally, there's a weaker sa sound at the end that again stimulates a few high-frequency sounds, high-frequency cells. So as you're listening to me now and throughout life, you have this constant jumping around of a pattern of different things being excited. It's been analogized to a piano in reverse, right? A piano takes individual pure tones and it combines them into a melodious whole. This thing takes the melodious whole and it undoes the work of the piano. It separates the different frequencies out and tells the brain about each one of them independently. And then it's up to the brain to decide what it's heard based on that spectrum of different sounds. Yeah, so the question is, why do we mishear words? And this is certainly one place the communication can break down. And let me point out a place that you will most commonly experience that, and that is with people who have hearing aids. With a hearing aid, one can make the sound louder, but as I showed you already, making the sound louder isn't so important as making the frequency sensitivity as good as possible. And when your hearing deteriorates, as I'll teach you in a minute, your ability to tell one frequency from another deteriorates. It's rather as if you have bad glasses, right? You can't read because things are blurry. Sounds blur in the same way. So your ability to tell the difference between whatever this is the word see, and this is the word sang, to tell those two apart, see, sang, it's obvious when I say sa and see and sang, it's obvious that they're different. But if you imagine sort of defocusing these farther, because the system is not able to attend to different frequencies so sharply, you can see why things begin to be confused. So the cells that do this are called hair cells, as I've said. The hair cells have given that name because at the top of each cell is this so-called hair bundle, this cluster of little filaments or hairs sticking out the top. This is about 25 microns tall, so that's 1/1000th of an inch. Here's the nucleus. It's not a nerve cell. It doesn't have axons, it doesn't have dendrites, but it does make synapses. So it sends chemical messages from the base of the cell to these nerve fibers that then carry information pop, pop, pop into the brain. So the specialization that's unique to the hair cell, this hair bundle, can be seen best by looking down on it from above. When you look at it in a scanning microscope, you can see this really elegant organ pipe arrangement of the so-called stereocilia. So stereocilia means stiff hair, and each of these is indeed a stiff hair filled with the protein actin. So actin is one of the proteins that also makes up our muscle fibers. You can see two important features of the hair bundle in this image. One is that these hairs are not all of the same length. There's always a short edge to the hair bundle, and then they get longer and longer as you go across. So they have this sort of organ pipe stacked look to them. And the other is that the hairs don't stand straight upright. They always heel over towards each other, so you get this conical or teepee-like structure. One final feature is there's one weird process, this guy here. This is a different one called the kinocilium. It has this ball on the end of it. And it turns out that this is where the inputs to the cell come. The stereocilia do the work, but mechanical inputs come to the kinocilium and are communicated from it to the rest of the hair bundle, and I'll show you how that occurs. If we cut across this hair bundle about halfway up, we'll see this very regular pattern. The stereocilia are in perfect hexagonal axes, so you can see 1, 2, 3 axes. The kinocilium is eccentrically located at the tall edge of the bundle, and you can see it's different inside. The stereocilia, as I said, are full of actin. This contains the protein tubulin, and it's rather like the motile cilia that you find in the bronchial epithelia or on the tail of sperm. So it's able to actually wiggle back and forth in a powered way. These other guys do not have that capacity in and of themselves. If we look at the base of a stereocilium, we find that it's characteristically tapered. In the last micrometer or so before it inserts into the top of the hair cell, it squeezes down from having about 500 actin filaments to having only about 20 actin filaments. And as a consequence, it's most compliant or most floppy there at the base. And you can see that if you take a living hair bundle such as this one and touch it with a glass probe such as this. Now the whole hair bundle moves as a unit, and that's because they're little filaments, protein fibers that hold the cilia together. But the individual cilia stay quite straight. So take a look at this longest stereocilium here, before and after bending. It's quite rigid. It simply pivots at its basal insertion. And that pivoting is important, because if you imagine that all these guys are pivoting at their bases, each pair of them must slide with respect to its neighbors. And that sliding at the tips turns out to be where the action is. That's where the transduction event itself takes place. So if we look at such a hair bundle, this is a model of the same thing, and you can see it's got about 60 of these stereocilia. Some of them are copper, some of them are aluminum. And when it moves, you can see them sliding with respect to each other at the tip. And so this will turn out to be the stimulus that excites the cell to give rise to an electrical response. In other words, the transduction event will involve the back and forth motion of this. Just to give you a little bit of an insight into what I'll tell you in a moment, one of the ways we study this is to mechanically stimulate the hair bundle with a flexible glass fiber that's very tiny. So this represents a flexible glass fiber that's very tiny. And what we do is to go down to bedrock under one of these buildings, as far away from subways and things as we can get, and then use a stimulator to apply a mechanical force to the hair bundle. So instead of playing a sound, we actually push on the hair bundle like that. And we can then measure how the hair bundle responds, or how the hair cell responds. First, we can measure how stiff the hair bundle is. And you can see how we're measuring the stiffness. This thing flexes a bit depending upon how much force I apply. So the more force I apply, the more it bends. This is just like measuring a fish, right? By weighing a fish by measuring how much the pole bends. So if we calibrate this thing, we can find out just exactly how stiff it is. It turns out a nice way of calibrating it is that if I stick this in water and just let it sit there and look at it with a very sensitive microscope, I find that it's moving around, it's jiggling. Now I'm jiggling because I'm scared, but under the microscope, what would be jiggling the tip of this? I heard somebody said it. Water, and what is happening with the water? What are water molecules doing? Yeah, exactly. Water molecules are bouncing off of it. This is what's called Brownian motion, after the guy who first found it. So this is going on all the time. In fact, what sets the limit on our hearing is the Brownian motion in the ear. We're able to hear everything down to the point at which the water molecules in our ear are banging against the cells. So this hair bundle will respond when I give a stimulus like that. It won't respond to a stimulus that's terribly tiny, simply because in reality, it's constantly doing that. It's constantly being jittered by the water molecules around it. So using such a probe, we can tell how stiff the hair bundle is, and I'll show you responses from it later. The other thing we can do is with an electrode, we can stick an electrode into the cell and measure its electrical responses. So we can measure directly what the hair cell is up to at any given time. So, okay, stay. Here's the sort of preparation we use. You can see under a microscope individual hair cells. You can see the hair bundle, the nice hexagonal packing, the canocilium on one edge. And we can then move the hair bundle back and forth as a function of time. So this represents the stimulus, back and forth and back and forth. And if we record an electrical signal from the cell at the same time, we see that the cell gets more positive, more negative, more positive, more negative. And as I've told you already, such signals in cells come about because ions carrying current flow across the membrane of the cell. And this is in fact no exception. There are exactly such ions flowing in this case. Notice that as the stimulus gets bigger and bigger, the response gets bigger and bigger until it saturates in both the positive and the negative direction. And the peak response is about 10 millivolts, that is 10,000ths of 1 volt. Now, because we're measuring the back and forth motion of the bundle and simultaneously measuring the electrical response, we can plot the one against the other, plot displacement and response, and we get curves like this. And the point to be made of this curve is that the response is very narrow. The cell goes from no response to a maximum response over a distance of only about 0.2 of a micrometer, which is a very, very small distance indeed. So a micrometer is, of course, a millionth of a meter, and this is 1/5 of that amount that is involved. So the system is very sensitive, and as I said, at threshold, it's sensitive to atomic dimensions. In addition, this sensitivity is directional. So if I move the hair bundle back and forth on its axis of symmetry, towards the canocilium or away from it, I get a maximum response. If I move it at a right angle, I get no response at all. And if I move it at other angles in between, I get a response that progressively declines. And in fact, if you do the geometry, it declines as the cosine of the angle. So what that means is if I have a stimulus like this one, there'll be a response to the cosine, which represents the portion of the stimulus along this axis, but no response to the component at a right angle to that. This is very valuable in our vestibular apparatus because it means each hair cell is most sensitive in one particular direction. So if I have a whole array of hair cells, whenever I make a movement, whatever direction I move, some cells will be excited, some cells will be inhibited, other cells won't care. And the brain can poll those cells, ask which ones were excited to what extent, and by that means it can determine exactly along what vector I was moving at a given instant. So where does the response originate? If we sort of sniff around the hair bundle while we're stimulating it, so in other words, we take an electrode that measures current and we move it around the hair bundle to different positions while we're stimulating, what we find somewhat surprisingly is the responses are small at the base. They're biggest up here at the beveled tip. So these things represent the responses. You can see these are much less than those. So somewhat surprisingly, it isn't down here where these stereocilia bend that the action occurs. It's up here where they touch each other, up here where the tips scrape one against the other. And in fact, there's a specialization at that point called the tip link. So you can see at the top of each shorter stereocilium, there's this fine braid of two proteins going up to the side of the next one. And here's another tip link up at this point here. So the evidence now suggests that these little strings pull on a particular type of ion channel and pop it open. So at the base of each of these, there seem to be 2 ion channels. And here's a diagram of what we think happens. When the hair cell's resting on the left, those little strings, the tip links, are fairly slack, and the channels are generally closed. When we push on the hair bundle, moving it to the right, the sliding between the adjacent stereocilia puts tension in the springs, and you can see the little red gates of the channels have popped open, And of course, ions will flow through those now, mostly potassium and calcium ions. Those will make the inside of the cell more positive and give rise to the electrical response. And that response is then passed on to the nerve fiber and so on into the brain. The next thing we can do is to measure mechanical responses from the hair bundle. And I will keep this slide that shows how we do it. As I've said, we attach a fine glass probe to the hair bundle. We can image the tip of that onto what's called a photodiode. That's a very sensitive apparatus for measuring the motion of the hair bundle. If the hair bundle is at rest, the fiber attached to it is straight. But if we move the base of the fiber a little bit, it will pull the hair bundle in one direction. But as you've already seen, the fiber will bend in the other direction by this amount. And knowing how much it's bent and how stiff the fiber is, we know how much force the hair bundle is producing. What we expect is something a little weird, which is that we can actually feel the opening and closing of these ion channels. And the rationale for this is shown here. This is a rat trap. It's been defanged so I don't lose a finger, but you can see it opens and closes. It has a gate, and it represents an ion channel that lets, say, potassium and sodium things flow through it. So it can be open or closed, and it has attached to it a tip link like the one I just showed you, the little piece of elastic. So imagine that this is one stereocilia and this is another. What I'm telling you is that these channels at the tips of the stereocilia open and close as the hair bundle moves back and forth in response to the sound of your voice. Now, if you came up here and pushed on my arms, you would actually feel this opening and closing. Because imagine, if you push on the hair bundle now, as the hair bundle moves to your right, that rubber band will extend, and you would feel that stiffness. And the stiffness is the same if the channel is closed or if the channel is open. But something interesting happens if the channel opens while I'm pushing. If I begin to push while the channel is closed and it opens, the rubber band all of a sudden goes slack until I apply more force. Okay? So we expect stiffness, less stiffness, and now more stiffness. We expect a soft spot in the response of the hair bundle to such deflections. And in fact, we measure that. If we push on the hair bundle, we see that it starts out being quite stiff. So this is force and this is displacement. The slope of that curve is stiffness. And we see that it starts out being stiff and then becomes abruptly less stiff. In fact, it becomes negatively stiff and then becomes ordinarily stiff again. Now, this is a violation of something you've learned about, quite probably, which is Hooke's law. Hooke's law says that there's a linear, straightforward relationship between displacement and force. So Hooke's law should be like that, right? Force is proportional to displacement. There's a straight line running through the origin. But here we have something that deviates wildly from that, and in fact has this weird negative stiffness in the middle. And just to show you what negative stiffness means, this next slide is a diagram. Ordinarily, when the bundle's at rest, the fiber attached to it is straight. If we move the base of the fiber a little bit, it will pull the hair bundle, sorry, I want to move a lot first, let's do that. Pull the hair bundle to the right a long distance, the fiber will bend back to the left. But if I instead move a small distance, we find that the fiber tip actually moves farther than the fiber base. So in other words, the hair bundle is pulling the fiber farther than the fiber is being asked to move. So it's a strange thing, right? It means that it's as if I come up to an object such as this table, whatever it is, the podium, if I push upon it, instead of pushing back, it instead pulls me in the same direction. It says that the hair bundle, when you push upon it at a certain distance, will actually pull farther in the same direction when the fiber is applied to it. In other words, the hair bundle has in it the capacity to do work. It has an energy source built into it. The reason that this comes about is a bit obscure. This diagram tries to explain it, but it's basically what I've already showed you with the trap. That as you begin to push on the system, it starts out with the channel shut. Now as you go, the channel's open and it becomes softer. As you go farther, it becomes stiff again. That corresponds to the different portions of this curve. At the outset, you have a system with the channels closed. These are the channels now at this point. First, one channel opens. When that one channel opens here, then there's more force on the other channels. They tend to open as well. Finally, all of the channels are open, and the system is unstable in between. When there's no force, it's stable here. When there's a lot of force, it's stable here. But in between, it jumps back and forth. It can't reside stably in the portion of the curve in between those two. So I'll show you in a minute why this matters. This turns out to underlie one of the ear's most remarkable characteristics. Also from this, we can measure things about the ear. We can measure that each hair bundle has about 100 channels. And because there are about 50 or 60 stereocilia, that means 2 on each of the stereocilia. We can measure how far the gate swings. We can measure how stiff this is. It turns out to be similar to the stiffness of a single protein molecule. And we can measure how tight is the structure that closes the channel. Another funny thing about this is that it explains an old phenomenon in music. So back in 1714, a great violinist, Giuseppe Tartini, noticed what he called phantom tones, and they've also been called difference tones or combination tones, and he described them. What he noticed is this: if you play 2 frequencies, and he was tuning his violin, so he was playing 2 strings at the same time. If he plays a high frequency F2 and a lower one F1, he would of course perceive those 2 sounds. He would perceive harmonics, so the second harmonic, the third harmonic, and so on. None of those is a surprise. The surprise is that he could hear frequencies like F2 plus or minus F1, or twice F2 plus or minus F1, twice F1 plus or minus F2. These are not harmonics. They're not tones that an ordinary violin string or something like that should be producing. In fact, he convinced himself they're being created in our ears. They're not being created by the instrument. I've already showed you the relationship between force and displacement is this funny curve with a kink in it. And those of you with the necessary math can appreciate that you could plot this as a relationship of force to the linear and the quadratic and the cubic terms of whatever x, whatever displacement you put in. If you put in 2 sine waves, you would get various combination tones of the sort that are observed. Specifically, the quadratic component would give you F2 plus or minus F1. The cubic component would give you these higher order terms, 2F1 plus or minus F2, and so on. And indeed, these things are generated by hair bundles. The theory I just showed you says that if we play these 2 frequencies, we would expect vibrations measured here at those frequencies, but also at these various other phantom tones. When we do this actual thing to a hair bundle, we see what we anticipate, namely oscillation at these other frequencies that are not being played. And I want to play you a demonstration of this to show you how strong the effect is. So in this demonstration, I'm going to play a pure single tone for 7 seconds. This is F1, the lower tone. Then I'll play a higher tone that descends, that's F2. Then I'll play the same 2 tones simultaneously. Now the strongest of these different tones is twice F1 minus F2. So twice F1 would be up here. Remember, this is not being played. But twice F1 take away F2 starts out as a small distance, and then it gets greater and greater. So 2F1 minus F2 starts out low and rises. And the reason for doing it this way is you'll hear a constant tone and a falling tone. But if you hear a rising tone, I'm not playing it, it's being generated in your ear. So here is the constant tone. Now the descending tone. And now the exact same 2 things played simultaneously, and listen for a rising tone, particularly towards the end. Can most of you hear it? I'll do it one more time, since you can all hear it. Oop. So I'm really and truly not playing that. I mean, you have to trust me on this one. It's strong enough that some composers have used this in creating music. So the first one was Ligeti, Hungarian composer, who composed a number of things in which the melody was played by no instrument, but instead 2 instruments would play a tone stream of other frequencies, and the 2F1 minus F2 would synthesize the music that he wanted people to hear. And more recently, Karl-Heinz Stockhausen has also produced music. Again, no melody is being played, but the melody is synthesized in one's own ear. Okay, so I said there was a really important reason we care about these nonlinear behaviors, that is the peculiar behavior of the hair bundle, and that is what's called the active process. The ear has something magical built into it. We've only begun to understand it within the last decade or two, and it was a big surprise because the things that it does are so weird. The active process has 4 characteristics. The first of them is amplification. So if we look at the vibration of the basilar membrane, just like the movie I showed you earlier in the talk, and we look at a particular place along the cochlea and play a range of frequencies, we find that a particular place will vibrate the most for one frequency. It'll vibrate less at a higher frequency, it'll vibrate less at a lower frequency. Of course, if we go to a different place, if we move towards the base of the cochlea, the vibrations will move to a higher frequency. If we move towards the apex, they'll move to a lower frequency. If we take the same animal and block its carotids, block the blood supply to its ear for just a few seconds, we find that this response goes away. We get just this plain blue curve. This is a logarithmic plot, and it's meant to suggest that this amplification is more than 2 orders of magnitude. That is, it's more than 100-fold. So our ears have a built-in hearing aid that turns sound up by more than 100-fold, sometimes as much as 1,000-fold. And it's working all the time. It's intrinsic. And in fact, when people become hard of hearing, what's happened is that their built-in hearing aid is simply blown out. So they have still responsive hair cells, but they've lost this intrinsic amplifier that we ordinarily take for granted. The second thing that happens is that they lose frequency selectivity. So ordinarily, each cell or each position along the basilar membrane is very sharply tuned to particular frequencies. If the animal does not have a blood supply, or if the animal is dead, the response is much broader. And even if you amplify the sound 100 times, you would get this very broad curve. And this is, in fact, what's wrong with hearing aids. If we've lost this sharp frequency discrimination, even if we make the sound 100 times louder, it's still very broad, and our ability to discriminate different speech sounds has deteriorated a great deal as a result. The third thing is called compressive nonlinearity. What that means is if we play a weak sound, we get a strong response at this position. If we make the sound 10 times as loud or 100 times as loud, the response does not grow in proportion, it grows less. If we do the same thing at a frequency away from the peak, like over here, then it grows in proportion. So somehow there's a compression of the response at the best frequency. And this again accounts for the fact that I told you that we can encompass such a broad range of intensities in our ear. 1,000,000-fold range in amplitude, 1,000,000,000,000-fold range in power. This is because, in fact, as the sound gets louder and louder, the response does not rise nearly as much. The final characteristic is the really weird one, and that's spontaneous otoacoustic emissions. Most of our ears will not only capture sound, but they will actually emit sound. So if we capture one of you and tie you up, take you, we have the rope, take you over to a sound chamber, the odds are 70% that one or both of your ears will emit sound in a suitably quiet environment. Maybe the ears will be different from each other, so the frequencies that come out will be characteristic of each ear. They stay constant over years unless your ear gets damaged, but your left ear will be different from your right ear, your ears will be different from my ears. It's quite an extraordinary thing. Now, and when this was first discovered, of course, nobody believed a word of it. It was so weird. There's not much use to having sound coming out of your ears. This is not considered behaviorally advantageous, but what's happening is that it's a feedback system. If you're in a loud environment, you don't need an amplifier. The system turns itself down. If you go into a quieter environment, the system gets progressively more and more sensitive. If you're in an ultra-quiet environment, home at night, alone, in the middle of the night, you can hear a pin drop. Your ear becomes much more sensitive. And if you go into a hyper-quiet environment like a sound chamber, the system turns itself up too far and it goes unstable. And it's just like the public address system. If we turned it up too far, it would go unstable and begin to howl. And that howling is, in fact, sound coming out of our ears. So the active process is so active that it can actually go unstable and begin to produce sounds that you can hear at a distance. all of these properties come, at least in part, from the activity of the hair bundles that I've already shown you. In other words, these hair bundles are not only recipients of sound vibrations, but they themselves can do active work. That active work enhances the sensitivity, and in the extreme case, it actually produces sound that comes back out as spontaneous otoacoustic emissions. So I'll briefly show you the evidence for that. If we apply a stimulus fiber to the hair bundle and move it back and forth, and I'm moving it plus or minus 10 nanometers, so a nanometer is a billionth of a meter, so again, a very small distance. You see that the response of the hair bundle is to oscillate at the same frequency as the stimulus, but to oscillate about twice as far. So there's an amplifier. The system is oscillating more than the stimulus that I'm giving it. And in fact, you can quantify this in some detail based on the theory of lawnmowers. Let's suppose you've got a lawnmower. And you have an old lawnmower of the kind that you've got to push actively. If you take that lawnmower, you can apply a certain force to it, and you go through a certain distance. So you're applying force to the right, and you move to the right. We know that force through distance is work. So we go to the right, we produce a force to the right, we go a certain distance. Now we push back to the left, the force is to your left, we're moving to your left, we come back along this trajectory. And the integral inside this of force and distance is work, or energy. So this is why you sweat, right? You've done this amount of work going around that circuit. Cool. Now you get a new lawnmower, and you're not so good with the new lawnmower. It's a power lawnmower. So you turn this thing on, but you turn it up too far. Now when you go to the right, you're pulling in the opposite direction as you go across. And similarly, you go back to the left, and when you get through with that, you were pulling to the left in the negative direction while moving in the positive direction. You were pulling to the right in the positive direction while moving in the negative direction. Okay? So this is, again, work. But in one case, I've had to do work on the lawnmower to get around the circuit. In the other case, the lawnmower has been doing work on me. And you notice the circulation is in opposite directions, right? That's how you can tell who's doing work, who's doing what to whom. So we can do the same thing to a hair bundle. We can apply mechanical forces to it and measure the work that it puts out to ask whether it really is capable of producing energy. And since some of you guys have had the physics, at any given time, shudder, right? At any given time, the forces at this point have to balance. So there's whatever external force I produce with the probe, there are the three forces of Newton's law, and there's whatever active force the hair bundle produces. All those things can be written out. It turns out that if you go over one cycle, one average cycle, two of the forces disappear. The elastic force disappears, where is it? And the inertial force disappears. And some of you already know that. If you take a rubber band, you apply force to a distance, you do work. But you can recover that work when you let it go back, right? In principle, you can get back, if it's a good rubber band, you can get back all the work you put in. The same with inertia. If I start a bowling ball this way, and then I run over there and decelerate it, I can recover the work that I put in accelerating in the first place. So you can just throw those terms out, turn the crank, and what's left is a simple relationship that whatever active work the hair bundle does has to be equal and opposite. The work that the force fiber does on the bundle and the work that's lost to drag, drag being viscosity, that is the drag of the water around the system. So we can do that, and I won't bother you with the whole issue, but the point is for individual hair bundles, we can again measure the work done, in this case by drag, the drag of the water on the hair bundle, in this case the work done by the fiber. And when I'm all finished, I can ask how much work is done on the aggregate. And the answer is that the hair bundle is doing active work. The active work is a total of what's called about 80 zeptojoules. Zepto is 10 to the minus 21, believe it or not. I mean, this sounds like Harpo and Groucho, right? But 10 to the minus 21 units of work. A very tiny amount, but a real amount of work. The hair bundle, in other words, really is capable of doing active work and accounting for the cochlear amplification. If we take the same hair bundle and let it run down, we find that as it runs down, it goes from producing active work here to producing no active work after it's dead, which just is showing that our accounting is honest. The other characteristics I'll cover very briefly. The hair bundle is tuned to a particular frequency. Here in a frog, this hair bundle was tuned to only 10 cycles per second, 10 hertz. The system produces compressive nonlinearity. Again, as the stimulus gets stronger and stronger, the sensitivity gets less and less. That is, each increment of stimulus produces less and less responsiveness. This allows us to compress signals. And finally, we get spontaneous oscillation. So I'll play you a little bit of a real spontaneous oscillation from a human. So you can hear that. Maybe you could turn it up a little bit. Good. Those high frequency pitches you can hear are these two frequencies. You can hear these lower ones a little bit. They're sort of hissing in the background. You can also hear a heartbeat and stomach gurgling. Right? So this is what the sound actually sounds like if you put your ear against another person's ear who's doing this, or put a sensitive microphone in. And indeed, individual hair bundles do this as well. The hair bundle, as you can see, is oscillating spontaneously. This one rather quickly and through a small amplitude. This one more slowly and through a large amplitude. Now these oscillations, it turns out, come in part from a myosin molecule. Myosin, remember, is the other protein involved in doing work in our muscles. And if we look at one of these hair bundles and then do chemical labeling for myosin, we find that there's a particular flavor of myosin that occurs up here at the top of the hair bundle where the tip links are. And in fact, we think that that myosin is occurring at the top of each tip link. It pulls on the links, opens and closes the channel, and causes the bundle to jerk back and forth. So such a system is capable of amplification. I won't go through the details in too extensively, but here's the basic idea. Imagine that we apply a sinusoidal stimulus, right? So we're going back and forth. Positive, negative, positive, negative. Each time we go positive, as shown by the green arrow, channels open, ions flow in, and among those ions is calcium. The calcium goes in, it binds to the channels and recloses them. We don't understand yet how it does. But as you can imagine, when it closes those channels, it's actually putting tension in those links. That tension pulls the hair bundle back to the left, which is the red arrow, and that stimulus, or that response helps accentuate the negative component of the stimulus. So in each cycle, calcium comes in, there's a move to the left. Calcium comes in, a move to the left. This turns out to power the system and give it amplification. And it works. It does what we anticipate. Namely, a small stimulus of a passive hair bundle produces a tiny response. But if we have this calcium binding and rebinding, we find that the response grows much bigger. In fact, it grows to be more than 100 times. It's tuned to a particular frequency. The tuning is best for small stimuli. It gets weaker and weaker and weaker with large stimuli, all the things we anticipate. And it finally undergoes what's called a Hopf bifurcation. So what is a bifurcation? A bifurcation is an abrupt qualitative change in the behavior of something as a result of a continuous change in a stimulus of some sort. So let's make a simple bifurcation. If I start with just a cylinder of paper, and I put it somewhere and I push down on it, what you would anticipate is up to a certain point, I will simply compress the paper. If it's elastic, it would get shorter and so on. But there'll be a bifurcation at some point in which it crimps, in which it abruptly folds. This is a different flavor of bifurcation. It's a bifurcation in which a system goes from being a stable amplifier to being an unstable one. So at the outset, a small stimulus gives a small response. If we go into a quieter room, we can turn the gain up. This gets bigger. It gets progressively bigger. And then finally, it goes unstable, and it begins to oscillate spontaneously. We now have spontaneous otoacoustic emissions coming out of the ear. I'll try to do a demonstration of this. The idea of the demonstration is I've got a very suspect circuit here that has a Hopf bifurcation in it. And if it works, what you will hear is when the system is near the bifurcation, the frequency, it will pick out one particular frequency and amplify that more than any other. So let's see what happens. If I say something, notice that it's sort of ping-y, ping, ping, ping. When I say the word ping, you hear that little oscillation afterwards. It's at a particular frequency. Tick, tock, ping, testing. And if I turn the knob here, it will get more and more ting, ping, 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 ping. We're approaching the bifurcation, and then we finally go across it, and it begins to oscillate spontaneously. Ping, 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 and then it goes unstable. So imagine now that you have, say, 3,000 sets of hair cells, each doing this. Each one is tuned to a different frequency. Each one of them can get as close to the bifurcation as it dares in order to get the best possible response. I think that's what's going on in your ear all the time. The system is constantly adjusting itself to optimality. If you're in a loud environment, you don't need it, the system turns itself down. But if you're in a quiet environment, this active process becomes prevalent. The active process takes the weak vibrations that would otherwise occur, it makes them much bigger, and it makes them much sharper, including an increase in our frequency resolution. So, to close, I want to turn to one last point, which is what happens when the system goes awry. I mean, I've said we have 10% of the population with hearing loss. What does that look like, and what are we doing about it? So, this is a normal human ear from a person who is unfortunately deceased with normal hearing. On the right, you see a human hair cell after aging and after the person has lost a lot of hearing. And you can see the regular pattern of the stereocilia has been grossly disrupted. Such a cell will typically die, and these cells in humans, and in fact in other mammals, are not mitotically replaced. So if you've lost your sensitivity to a particular frequency right now, you've lost it for good. It cannot be recovered by replacement of these cells. But the hope is, of course, that we'll find ways to change that. A number of groups are working on this. You'll see in today's paper the first report of an attempt to use a virus containing a particular human gene inserted into a human ear to try to replace missing hair cells. So this is exciting. It's the first time it's been reported in humans. Our particular work is done with the zebrafish. These are little dime store fish that many of you know, and they have the advantage of being very small, easy to keep in large numbers. So they're the sort of vertebrate equivalent of the fruit flies that Vanessa and her colleagues work with. We have 10,000 of them in a room that's not as big as this stage, so they're easy to maintain in large numbers. Excuse me. The larval zebrafish at about 1 week of age is about 4 or 5 millimeters long, and it has hair cells along its side. The zebrafish uses those hair cells to measure water motions. So it can measure its flow through the water, it can measure the flow of water around it, it can feel predators coming, it can sense prey and strike at them. The nice thing from an experimental point of view is we can label those cells with fluorescent dyes, as we've done here. We can see them very nicely along the length of the tail, and we can examine them at higher power in the microscope. Each of these little clusters of hair cells is called a neuromast. There are hair cells, such as these two red ones, that are sensitive to motion to the right. Remember, pushing towards the tall edge of the hair bundle depolarizes or excites the cell. These two blue cells, on the contrary, are sensitive to motion to the left. So as this fish moves through the water, the back and forth motion of its kinocilia and its stereocilia will tell it which direction it's moving, how fast it's moving, and so on. If we look in the scanning microscope, we see that there are miniature hair bundles at the bottom of each of these. These thick things are the kinocilia. Here are the stereocilia at the base, and you can see half of them are facing to your right, half of them are facing to your left. The way in which this organ is formed is rather neat. The organ actually originates just behind the ear, and then there's a migration along the tail, and as this thing migrates, it drops off clusters of cells that are going to become these sensory receptors called neuromasts. So here's a time-lapse film showing this migration. Can you bring the lights down a bit? This is the so-called primordium, and as it moves, it drops off one cluster of hair cells, now a second cluster of hair cells, a third cluster of hair cells, and so on. It keeps on going down towards the tail. So this represents about 8 hours during the first couple of days of a zebrafish's life. If we then focus in on one of these clusters of cells, we'll see new hair cells being formed. So each of these bright things is a pair of hair cells, and I want to call your attention to this dark cell here at the bottom, which starts out quite black. These hair cells are labeled with something called green fluorescent protein, which enables us to see hair cells specifically. Here you can see a new pair of hair cells that are formed at the top. Notice that this cell is now turning green. It's decided it wants to be a hair cell. It's going to swell, and in just a minute, after a very excited macrophage moves through, it'll undergo a division. Watch it divide. Pop, okay, a mitosis into 2 cells. So those two will become hair cells. This cell will blow up. So these cells are turning over all the time in the zebrafish. Unlike ourselves, they can regenerate as many new hair cells as pairs as they ever want to replace the ones that are being worn out and are falling out of the animal. So if we look at different stages, we find initially they're just 2 hair cells, then there are 2 adult pairs, a younger pair. Here are 4 adult pairs. Notice that they're facing in opposite directions. These are the kinocilia. Half of them face left, half of them face right. Here there are 6 cells on each side, and so on. So in this organ, which is very nice for research, there are stem cells, cells that replace themselves and also produce another kind of cell that divides and makes 2 baby hair cells. They mature, they age, they get too old, they finally die at this position. So the North Pole and the South Pole are constantly making new ones, and they're dying here along the equator. So this is a nice experimental system because we can put in fluorescent labels that enable us to isolate these different cells and ask what genes each of them is expressing. And of course, our hope is that we will identify genes that are being turned on during these stages, which we can also activate in humans. Imagine that we could go back to a human ear and turn on whatever makes this step occur. We would then have a cell that regenerates itself and now produces 2 new hair cells. So that's the goal of this research. In the meantime, there's another modality that's been developed for dealing with hearing loss that's been incredibly successful, and this is the cochlear prosthesis, the artificial ear. And the idea of that is based on something you've already heard about. Remember, when the ear is working normally, you have 16,000 hair cells, each of them sensitive to a particular frequency. You have nerve fibers connected with them. Now here's an individual in which all of those hair cells have been killed, maybe by use of a drug that was necessary to save his or her life. What are you going to do? Well, what you can imagine doing is stimulating the nerve fibers directly. Because if these nerve fibers are stimulated, the impulses going into the brain will look no different in the brain from the nerve fibers that were activated when the normal sound was heard. So you could potentially fool the brain, and that's what the cochlear prosthesis does. The prosthesis consists of a small device which picks up sound. It used to be the size of a cigarette pack or the like and fit in a pocket. Now it's so small it can fit on the frames of eyeglasses or the like. So you simply are wearing it all the time. Maybe some of you here have a cochlear prosthesis. This device picks up the sound, it splits it into multiple channels, just as the cochlea normally should. And contemporary devices use about 20 separate channels. The device then uses a magnetic antenna to send signals across the skin to the interior electrode. And the advantage of the magnetic stimulation is that there are no wires going through the skin, no way for bacteria to get in, no way for infections to occur. Then inside the ear, there is a coiled prosthesis, a structure like this. Whoops, sorry. I got one ahead of myself. We should have, yeah. This coil structure, which is inserted into the bottom turn of the cochlea. And as you can see, it consists of pairs of electrodes here, 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 and so on. So each pair of electrodes corresponds to a different frequency. This is a really old one that has only 8 electrode pairs, but I like it just because it's easier to see what's going on. So if you imagine you pass current between 2 of these, it will stimulate any nearby nerve fibers. And then the device is rigged so that the ones nearest the base, which would be these, are used whenever a high frequency comes in. The ones nearest the tip, which are these pair, are used for the lowest frequencies. And what I'll try to play you is a demonstration in which I've taken a piece of human speech, but I've thrown away all the information, except in the first case, just 5 frequencies. This will sound horrible, so you're not expected to get much out of it, but let's just see how it sounds. Okay, I defy anybody to make sense of that. The very first ones that were used were even more primitive than that, and people really liked them. I mean, for deaf people, even having this crude thing was very valuable. There's some sounds, for example, that you can't see by lip reading because they're made at the back of the mouth, the back of the throat. And you can get a sense from this very crude device of the presence of those sounds, cuz and thus, things like that. And for even the very first ones that were created were immediately enthusiastically adopted by people. Now I'll play you 10 frequencies at the same time, and you may get a word or two. You might up the level just a touch.
Cochlear prosthesis is now in everyday use by nearly
So who got any word? Cochlear prosthesis? 100, have you heard this before? Really? You get a t-shirt. Nobody's ever got that much before. You want a t-shirt? Or where are you? I can't see you. Somebody pass this back to the lucky winner. That's very good. So I'll now play 20 channels, and 20 channels is about what contemporary devices have, and you may get more of it.
The cochlear prosthesis is now in everyday use by nearly 100,000 people worldwide.
So what do you hear? 100,000 people. So here's the cochlear prosthesis is now in everyday use by nearly 100,000 people worldwide. So two cool things. One is it's now 300,000 people worldwide. So they are so successful that there are many people, 300,000, who were totally deaf, who can hear again as a result. The other cool thing is this. I'll play the 20 channel again, and you cannot not understand it. In other words, your brain, having heard it once, can immediately understand it perfectly.
The cochlear prosthesis is now in everyday use by nearly 100,000 people worldwide.
Isn't that weird? And this actually happened when the first patients were operated. I was in San Francisco when another group was doing this in San Francisco. The first person whom they got who had had good speech and lost her hearing was an Irish lady who flew in from Ireland. And she had the surgery and afterwards came out of the anesthetic, and people tried to speak with her, and she didn't comprehend anything, and everybody was very depressed. And then her son came in and said, "Oh, how are you doing?" And she immediately started talking fantastically in Irish. So she just didn't understand the San Francisco accent like man. So people just get up off the operating table and immediately have remarkable recovery with these. So some of you may have these, some of you will get these. The hope is that the cochlear prosthesis will be a way of dealing with the rash of hearing problems over the next few decades and during the time when development of longer-term therapies, such as restoration of the hair cells, becomes possible. So I'll stop at this point, and we'll take some more questions. Thank you very much. And I should say, if you don't have good questions for us, then we'll have some questions for you. So Vanessa and I are allowed to ask questions as well.
Before you were showing the cochlear prosthesis, you talked about stem cells. What about how long do you think it'll take, and will it be possible to repair like senses or any other things that don't work with the use of stem cell?
It's always a guessing game because there's so many things that can go right and can go wrong that we don't know. But that is certainly a viable option. It's being tried now, particularly in the visual system. There are particular conditions in which the photoreceptor cells that I described earlier will undergo degeneration. People are trying to restore those through the use of stem cells. And I don't know the current state of that. I think it's progressed to the point of trying it in humans. But whether there's any success yet, I can't tell you. The hope in hearing is very similar. We would prefer, rather than having to get stem cells and inject them, we would prefer to take the strategy that was in today's paper of putting in viruses or the like with so-called transcription factors that are molecular switches that turn genes on or off. We would prefer to do that simply because what would regenerate would be the patient's own cells. And so without having to do surgery or have anything in a dish or use any foreign cells, you would just turn your own cells back on, have them make one more set of hair cells and then shut up, which is what you want. Whether that can be done or not, we don't know. And it might be necessary to put them in a dish, coax them along with various chemicals, and then re-inject them.
Okay, hi. So as teenagers, since we listen to extremely loud music, which probably is not good for us, what part of our inner ear gets damaged when we do this to ourselves?
Yeah, so it is the hair cells. I mean, 95% or more of all the hearing problems come from the hair cells, and that's why we're so keen to regenerate them. We tend to lose particularly the higher frequencies. If we make a diagram of our sensitivity to different frequencies, To start with, this would be 20 hertz. This would be 20 kilohertz. So these would be very high frequencies. And this would be how sensitive you are. Human hearing normally looks sort of like that. We're sensitive at middle frequencies, most sensitive around 4 kilohertz, not very sensitive at very low or very high frequencies. With aging and most exposure to sounds, what happens is this is whittled away. You progressively lose the high frequencies. So I'm down to 12 kilohertz or something like that and falling. So far, as you saw from the sonograph, it doesn't matter too much because except for my wife, who has a high-pitched voice, I can hear everything. But you know, by the time you get down to 8 kilohertz or something like that, it really gets to be an impediment. And as you can see, what you tend to lose are the consonants, the th, tus, puz. Some languages, tonal languages like Mandarin and certain other languages, are less sensitive to this because they use tones which are down at a lower frequency range. They use less, or they place less importance on the high frequency consonants. So why these cells wear out first is also something we don't understand very well. Obviously, these cells are working much harder. Here, the hair bundle is actually going back and forth 20,000 times per second. These guys down here have bigger hair bundles, and they're going back and forth 20 times a second. So these seem simply to wear out, but exactly at a molecular level what that means, we still don't understand.
Doctor, what causes ringing in the ear?
Oh yeah, good point. So that's very common. There are many of us in this room who have tinnitus. And first of all, notice it's tinnitus, not tinnitus. This represents an inflammation. So if you have conjunctivitis or kneitis or brainitis or whatever, encephalitis, this is not an inflammation. So I mention this just because many people see that ending. They think it's itis instead of itis. Never mind. So it is an inflammation. Tinnitus means ringing in the ears. And it can vary widely in its characteristics. So some tinnitus is what's called bell-like, low-frequency tones. Most of it is high pitch. And it comes from a number of different sources. First of all, there are certain drugs that cause it, particularly aspirin and quinine. So if you're taking antimalarials, you can have it from the quinine or quinine derivatives. It used to be particularly when a lot of aspirin was used for arthritis, that people who were little old ladies who were taking 4 or 5 grams a day often got tinnitus from that. These days, people use non-steroidal things, ibuprofen and so on, that don't have this effect, so that has more or less vanished. Another thing that can cause it is a particular kind of tumor. This is called an acoustic neuroma. And this is why the doctor, after she's finished hitting you with a hammer, will ask if you have ringing in the ears. If you have one of these tumors, particularly on one side, it tends to cause one-sided, relatively low-frequency tinnitus. So, you know, anytime you have low-frequency, single-sided tinnitus, you immediately want to do an MRI or something to make sure this tumor is not there. The tumor is benign, but it occurs in a bad place at the base of the brain. If it grows, it presses on the respiration center, the heartbeat center, all the centers, you know, favorite centers. So it has to be taken out. Most tinnitus is idiopathic. Idiopathic is Latin for beats me. We don't really understand it, except that it seems to be like phantom limb pain. As you probably know, if you have an amputation, people who have amputated a hand or an arm often will continue to feel pain or flexion or discomfort in the missing part of their body. And what is known to happen in that case is as a result of the lack of neural signals coming from the missing piece, the part of the somatosensory or body cortex that ordinarily gets that input isn't getting any input. It turns itself up. It tries to become more sensitive until it goes unstable and it starts feeling things that aren't really there. The same thing seems to happen with tinnitus. If we've lost our hearing in a particular range, so in my case, wherever it went, having lost these higher frequencies, I have tinnitus along in here somewhere. I constantly hear these high frequencies to which I'm no longer sensitive. In my case, I got hit by a soccer ball when I was in 11th grade, and that damaged some hair cells, and this ear has been ringing like a bell ever since. Tinnitus can be a real problem. I mean, mine fortunately has not progressed, and you know, I just ignore it. But some people can't. I mean, some people, it keeps them awake at night, it keeps them from concentrating, and it can lead actually to depression and suicide. One thing that is good news is you can somewhat mask it just by playing white noise. So psh types of noises played at night or when one is concentrating will do a job basically just turning the whole nervous system sensitivity down. The tinnitus also has less of an effect. But for example, with music, that's a problem. You can't listen to any music while going psh. So this interferes with some quality of life issues. So anyway, it's something people are working on, but we do not know the full answer.
So this question pertains to the first lecture. If that's all right.
Sure. I vaguely remember it.
You're welcome. So I know certain people, when they're a child, they may hate a particular food. Then as they get older, they may start to love it. Is there any particular reason for this?
Here's our taste expert.
I mean, many of our preferences, as we discussed earlier, many of our, for example, our ability to recognize and discriminate different tastes and smells is actually dependent on learning, and so these associations can be very strong. And so simple taste preferences, there are a lot of them that are actually very much ingrained culturally just from experience. So you may learn to associate, you know, and I think your taste repertoire actually changes as you progress as well, the different types of receptors that are available. But a lot of it is based on experience and learning.
There are negative preferences that way as well. There's a very well-known thing called the Garcia phenomenon that most of us have had the pleasure of experiencing, which is if you eat bad food of some sort, you then develop a terrific distaste for whatever you last ate. And many people will swear entirely off of some food, even though they know there's nothing wrong with sushi or whatever it happens to be. Having been sick once, they are chastened about doing it again. And obviously this has a selective advantage. In the wild, if you eat a certain berry and it makes you almost dead, it's just as well to stop at that point.
The phenomenon of vertigo?
Yes, okay. So remember I mentioned the semicircular canals. The semicircular canals, so there are 3 of them on each side. They're mirror symmetric, and they give us our sensitivity to angular acceleration. Now, that information goes into the brain. I seem to be losing pens right and left. Here's the brain. I'm simplifying slightly. So the information from both sides converges on both halves of the brain. You have information from the right and the left labyrinth. The problem with vertigo comes about if you have, say, an infection in one of these. If you have an infection in one of these, you may start getting false nerve signals that are produced by the inflammation itself. Now the brain is in a real pickle, because on the one hand, this semicircular canal says I'm stationary. The other one is firing like mad, which says I'm rotating rapidly to my left. So what does the brain do? It hurls. I mean, the brain does not know how to reconcile these two things. You get violently ill. This is a real problem for some people with what's called Meniere's disease, which I can almost spell, but I don't get the French accents right. But anyway, it's Menieres. But professionals can put the 2 accents on. Meniere's disease is a disease in which there is a spontaneous swelling in the labyrinth, and often a person will get this on one side or the other, and he or she can sometimes just abruptly, while walking along, just suddenly keel over because the pressure suddenly gives a very strong acceleration stimulus on one side, and the imbalance between the two causes this to occur.
Does the installation of the cochlear implant make it difficult or impossible if a stem cell therapy later on never happens?
We think not. The reason we think not is that sometimes, particularly in the first devices that were installed, the cochlear prosthesis failed. It may be that some wires broke, that the power supply was deficient, whatever. And it's then been necessary to re-implant those people. And that has worked quite successfully. So it seems that even though when the device is put in, it's somewhat invasive, you can take it out altogether, put in a second one, and it works okay. That strongly suggests if you took it out and replaced it with stem cell therapy, things would also be all right. So, so far there's no demonstration of that because there isn't yet a demonstration of stem cell therapy in humans in this context, but it looks very promising. This is also has bearing on when you should put in such implants in young children. If you're in a family in which there's known genetic hearing loss, or at birth you find your child is deaf, how long should you wait before inserting a prosthesis if you choose to do that? We know that there are critical periods for development of different senses and also for language. So the sooner we start speaking a language, the better we do at it. And most of us are incapable of learning an accentless language after about age 12 or 13. So it's desirable that kids start using their speech faculty as early as possible. It's now the national government policy that you can implant kids as young as 1 year of age. There are tests being done at still younger kids, under experimental protocols down to 6 months, to see if after 5 years or 10 years, there's an improvement in their status. But if a family decides that they want a prosthesis, it can now routinely be done at 1 year of age.
We have olfactory stem cells in our olfactory system, so why do you think that is the only sense that has stem cells as opposed to everything else?
Yeah, it's funny. We have no idea. Of course, all these senses have evolved independently of each other. So there are historical reasons for the way that different senses cells look, the way in which they originate, the way in which they reproduce, and so on. And I can only cite that it's simply an historical fact in the evolution of the system. But there's nothing intrinsic about it. If you just look at the cells, you can't say, gee, this one should be able to regenerate and that one shouldn't. Just a fact.
So I just want to thank you all for coming today, and I'd also really like to thank Jim for 2 very incredible presentations.